Hello and welcome to Sleepy Science. I'm so glad you're here. Tonight, we'll drift through the hidden networks that quietly hold life together, from bacteria that vote before they act, and cities of biofilm that pave their own streets, to slime molds that trace perfect routes, and ants and bees that leave maps in scent and dance. We'll wander up the scales, neural microcolumns and astrocyte waves that pass messages without words, immune swarms that circle danger, tissues that sculpt themselves with simple rules and morphogen slopes, genes that flip fate like careful switches, proteins whose shapes clasp like hands, and the cell's own highways and tense cables that give form its strength. Then we'll step into negotiations at the edge of life as viruses and hosts revise their playbooks. Feel ecosystems breathe in predator-prey rhythms and food webs loop energy home, and watch ideas spread across people as surely as currents flow through towns that eat, drink, and exhale. We'll follow the world's traffic, trade that stitches civilizations, grids that hum in balance, hubs where one delay ripples outward, and the internet's surprising geometry before peeking behind every product's hidden links and the markets where confidence is a fuse. We'll settle by a dish of living circuits learning, meet the guilds in your gut, trace voltage maps that nudge limbs into shape, listen to chemicals speaking in gradients, and finally let small robots borrow nature's rules to work as one. Tell me where you're watching from and what time it is there. If these unhurried explorations help you rest, please like, subscribe, and share. Now take a long breath, let your shoulders drop, and settle in where curiosity glows and the night teaches gently. Biofilms as cities, streets, waste, and defense. You think of slime and scum when you hear biofilm, but imagine you're drifting over a hidden city that builds itself without drawings or orders. A colony of microbes settles on a surface. Your tooth enamel before breakfast, a catheter wall in a hospital, a rock in a stream, and begins to weave a soft scaffold around itself until the slick becomes architecture, complete with streets for water, courtyards for sharing food, and thick walls that blunt almost everything the outside world throws at it. That is the invitation to look closer, because in this living city, the concrete is a gel called the extracellular matrix, mostly sugars braided with proteins, lipids, and strands of DNA that cells release like rebar. And once that mesh forms, the world inside no longer resembles free-swimming life. First comes attachment. At the beginning, it's tentative, a reversible brush of surface structures that feel the chemistry and texture underfoot. Then commitment, as cells exude sticky polymers and settle into place. From there, layers rise, not as a uniform carpet, but as uneven neighborhoods shaped by flow, nutrients, and chance. And thin water channels open like alleys, carrying oxygen and food inward and washing wastes outward so the interior doesn't choke. You learn quickly that this layout creates gradients. Oxygen richest near the surface, fading with depth, sugars and acids waxing and waning by micrometers. So different pockets of cells switch into different metabolic gears. The top layer may burn oxygen fast, the middle barter organic acids. The bottom run on fermentation or even use nitrates when air is gone. The patchwork turns a once simple population into a division of labor, which makes the whole far harder to starve. The walls themselves do more than hold shape. Their tangled sugars can bind antibiotics before those drugs reach their targets. Their proteins can soak up antimicrobial peptides like a sponge, and their DNA crosslinks with ions such as calcium to stiffen the gel, so the city becomes mechanically tough. Even if a drug slips through, many residents deep inside are growing slowly, and slow cells are notoriously hard to poison because most medicines target fast processes. Some residents duck into special states, persisters, where they ride out stress with minimal activity, not as mutants but as temporary survivors. When the storm passes, they wake and repopulate, which is why a treatment that looks like it worked on day 3 can fail on day 7. The shapes you can't see with the naked eye matter. In gentle flow, biofilms form bulbous mushrooms with stalks that let fluids circulate underneath. In fast flow, filmy streamers flutter from edges and can tangle into clogs that suddenly block pipes, a tiny cause with big consequences in medical lines and industrial systems. The matrix isn't just sugar mush either. Some bacteria lace it with protein fibers that behave like microscopic cables. Others extrude amyloid threads that harden like scaffolding. And many species borrow extracellular DNA not only as glue, but as a shared library. Cells pressed close together can swap genes by direct contact or pick up loose fragments, accelerating the spread of traits like metal resistance or new metabolic tricks. If you want examples you can feel with your tongue, dental plaque is a biofilm whose traffic rules influence cavities and gum disease. Bacteria ferment sugars into acids near the tooth surface. Local pH dips, minerals leach from enamel, and the longer the city sits undisturbed, the more its chemistry tilts against you. A brush or floss doesn't kill a city so much as bulldoze its streets, breaking the structure so saliva and oxygen can reset the terrain. In hospitals, the stakes are harsher. Biofilms that establish on implants, prostheses, and catheters can shrug off doses of antibiotics that would easily stop the same species floating freely. Immune cells struggle too, because the matrix blocks access, and some residents secrete detergents and enzymes that blunt attacks, while the geometry itself creates refuges where defenders cannot reach effectively. None of this requires you to believe in secret intentions. Just remember that life changes when it stops drifting and starts building. Once surfaces and gels exist, physics and chemistry carve the space into niches, and niches invite strategy. What works against these cities then? In nature, you find organisms that release enzymes to cut the matrix, nucleuses that snip stray DNA, glycosidases that shave off sugars, even specialized molecules that loosen crosslinks, so the walls soften and cells dislodge. In medicine and industry, similar tools become therapies and maintenance routines, often paired with antimicrobials, so that once the streets open and residents scatter, the drugs finally meet their targets. Surface design helps too. Coatings that resist attachment, textures that make it hard to anchor, materials that present water-loving brushes or neutral charges so the first foothold never sticks. When anchoring is delayed, the city never gets its base layer and the rest cannot rise. Water flow and cleaning schedules become urban planning. Too gentle, and you let streamers grow. Too rare, and you allow towers to thicken into fortresses. Consistent shear and timely demolition keep the map flat enough that ordinary sanitation works. Even viruses that prey on bacteria, phages, can thread through channels and crack defenses from within, especially when chosen to match the target residents. Combinations sometimes matter more than single silver bullets, because a city is not a single structure, but a set of functions that compensate for each other. The question for you, listening in the quiet, is not to fear what is small, but to respect what emerges when small things organize. A biofilm is proof that protection, logistics, and waste management can arise from simple building blocks as soon as space and time allow them to settle. That recognition teaches you to look for structure where you once saw sludge, to ask whether a stubborn problem is being held in place by hidden architecture rather than by obvious enemies, and to imagine solutions that change the city rather than just shouting at its citizens. And when you finish this, you might clean your teeth a little longer, rethink how a hospital tube is maintained, or picture a river rock as a bustling neighborhood. Because the truth is interesting enough without embellishment. Streets are real, waste has routes, and defense lives in the walls. Slime mold routing, nature's best network engineer. If a brainless sheet of living jelly can sketch a map that rivals a human engineer's, what else have you been underestimating about how intelligence can look? Meet the slime mold, especially Physarum polycephalum, a single giant cell with many nuclei that spreads as a thin, yellow organism across damp surfaces and quietly builds a transport network out of living tubes, not by thinking in symbols but by feeling the world and updating its own body in response. And the first time you watch it under time-lapse, you see a soft pulse sweep through its veins every minute or two. A peristaltic wave that pushes cytoplasm back and forth so nutrients slosh along like tides, and the passages that carry more flow slowly thicken, while the skinny dead ends fade until a sparse, elegant scaffold appears that reaches food sources efficiently, survives damage with just enough backup loops, and uses far less material than you expect. The secret is not a hidden mind but a set of physical rules. Rhythmic contractions powered by actin and myosin send fluid through a web of protoplasmic tubes. Flow strengthens the tubes that serve it, and the whole network prunes itself into a cost-effective layout with a small number of safety detours, a balance that engineers call the trade-off between efficiency and robustness. In classic experiments, researchers pinned oat flakes on a plate to mimic cities around Tokyo, and let the organism forage from a downtown in the middle. Within hours, the slime molded a pattern of connections that mirrored key links of the real rail network. Not perfectly, but recognizably, favoring short, reliable paths and preserving alternative routes when a single break would have stranded a region. And when the environment changed, adding light to discourage growth or drying a corridor, the layout morphed, rerouting around hazards while still aiming for low travel cost, like a living version of a planning algorithm that keeps running after the plan leaves the whiteboard. The same body as calculator trick solves mazes. Put the organism at one entrance, place food at the exit, make the walls unfriendly with light or salt, and it will spread in all directions at first, then gradually retract from punished zones and reinforce the corridor that actually leads to dinner until only that path remains. There is no inner diagram, only a feedback loop linking sensation, flow, and structure that accumulates experience in the shape of the body itself, a kind of morphological memory, where yesterday's good route is literally thicker today. So the next pulse follows it more strongly. The organism's senses are simple but enough. It prefers shade and moisture. It grows toward attractive chemicals drifting from food. It avoids bright light and certain salts, and because it is one continuous cell, local changes propagate as traveling waves that coordinate distant regions. So, an attractive flake on the left can tug on a branch growing on the right by altering rhythms across the whole sheet. Cut the network and it heals, first by clotting the breach and then by rebuilding tubes around the gap. And if two individuals meet, they can fuse and share their internal streams. A reminder that boundaries in biology are often softer than your vocabulary suggests. What makes this more than a curiosity is that the rules are general enough to inspire tools outside the petri dish. City planners and computer scientists have borrowed the mold's style to design roadway and pipeline layouts that minimize cost, while staying tolerant to disruption. Evacuation studies use its tendency to keep backup loops to test how bottlenecks form. Electronics researchers look at its habit of reinforcing high-traffic routes to think about reconfigurable circuits that route around faults without central control. Even farmers and foresters can read lessons about how resources and obstacles shape branching patterns in roots and fungal mycelia, which face similar decisions about where to invest growth for the best return tomorrow. The organism is not magic, so it is worth keeping the story honest. It does not always find the mathematically perfect solution, and in very complicated maps or hostile conditions, it can stall or wander. What it does with striking reliability is discover good enough designs quickly and adapt them when the ground rules change, which is exactly what most real systems need. It also shows hints of simple learning without any neurons at all. If you train a slime mold to cross a shallow barrier laced with a mild repellent to reach food, it hesitates at first but crosses faster over repeated trials, and the tolerance can persist for a while after the repellent is removed. In some studies, fusing a trained individual with an untrained one passed the habit along, suggesting that memory can, at least briefly, be encoded in circulating factors and the mechanical state of the network rather than in synapses. And whether or not you call that learning, it broadens your sense of where experience can live. If you want to apply these ideas to your own decisions, start small and physical the way the mold does. Explore widely to sample options, then let real-world feedback thicken the paths that truly feed you and allow the rest to wither. Keep a few loops so a single failure doesn't strand you. Make sure your sensors are honest. Light where you don't want to go, nourishment where you do, and let repeated rhythms carry information from one part of your life to another so that what you discover in one corner can guide growth elsewhere. There is a quiet comfort in this organism's pace. It pulses, it listens, it redraws, and over hours the messy sprawl becomes a map that works, not through willpower, but through patient sensitivity to what helps and what harms. And when you finally imagine the dish going dark and the organism settling into a steady hum of contractions, you can feel the lesson land gently. Sometimes the smartest plan is not to overthink, but to give your body and environment a chance to talk to each other until the best route thickens under its own weight. A living network teaching you that clarity can emerge from motion, and that a good path is less a stroke of genius than a shape that survives the night. And pheromone algorithms, shortest paths, without math. Imagine you dropped a line of sugar grains across a kitchen tile and invited a colony of ants to solve a puzzle you didn't tell them existed. two routes to the same reward, one a little shorter, one a little longer, both invisible to any single insect's mind. You watch a few scouts wander, apparently aimless, until one stumbles on the food and hurries home, brushing its belly to the ground as if signing its name, laying a faint chemical trace. And then the show begins, because other workers bump that trail with their antennae and choose to follow it. And as they do, they lay their own traces, and the path thickens, and soon a dark, shimmering line of bodies flows between nest and sugar like a living conveyor. And here's the twist that matters. If there are two bridges to the food, the shorter route fills and refills a little faster, so its chemical mark grows stronger a little earlier, and that tiny timing edge snowballs into dominance, even though no ant measures length or angles, producing what looks like a smart choice without a single calculation. This is a pheromone algorithm written in motion, a kind of problem-solving called stigmergy, where traces in the environment tell each individual where to go next, and the memory lives in the world rather than inside the brain. Development comes from details that aren't obvious until you slow your eyes. The signal is not permanent ink, but a perfume that evaporates. And that fading is not a flaw but a feature, because it prevents the colony from getting locked into yesterday's answer. If the snack is gone, or the path gets blocked, unused stretches cool off chemically and traffic wanes, while fresh explorations sprinkle new possibilities that, if they pay off, gather their own strength. Different species tune this balance in different ways. Fast evaporating trails encourage constant updating. Long-lasting ones stabilize profitable highways. And some ants, like the pharaoh ant, even leave no-entry chemicals that say, "Don't bother here," a negative mark that keeps crowds from banging into dead ends. Desert species add solar and skyline cues to stay oriented while still obeying the chemical hints. And leafcutters run heavy supply lines where pheromone is the backbone, but traffic rules and lane formation help them avoid gridlock. Because outbound workers carrying nothing and inbound workers carrying leaf discs change their step timing when they collide, spontaneously forming separate lanes like patient commuters who never met a traffic engineer. The physics of bodies and scent becomes logic. Antennae scan left and right like tiny divining rods, and a worker's chance of turning increases if one side smells richer than the other. So a gradient of scent becomes a decision at a fork. Bumps between ants aren't noise, but signals too. Repeated contacts with successful foragers increase the odds of joining the flow. So inspiration spreads by touch, while the road itself becomes a guide. In two-bridge experiments, even very slight differences in length or roughness get amplified, because ants on the better bridge complete trips faster, lay more fresh scent per minute, and pull more recruits their way. A feedback loop checked by evaporation that keeps the colony from following a ghost forever. None of this requires grand claims about perfection, and honesty makes it more impressive. In messy terrains with shifting risks, ants don't always find the shortest route, but they consistently find a short and reliable one quickly and then keep it flexible. If you drop an obstacle onto their boulevard, some workers peel off around the barrier, exploring both sides at once, and the first detour that succeeds gets copied and strengthened. The other withers. And if you remove the obstacle later, the old highway can reappear because a trickle of wanderers never stop testing alternatives. The colony mixes exploitation and exploration without ever naming the strategy. And that balance is what keeps food arriving even when the map changes. The chemistry behind all this is a family, not a single molecule. Alarm signals that trigger scattering and defense, trail signals tuned for recruitment, cuticular blends that label nestmates, and even graveyard markers that keep the living from obsessing over the dead. A worker's behavior depends on which bottle in nature's cabinet just opened, and context matters. Hunger level, time of day, crowding at the source. So the algorithm is not a rigid script, but a set of rules that lean toward good choices when the world cooperates and still offer escape routes when it doesn't. Sometimes a species recruits by guiding a single follower body to body in a slow tandem run, laying little or no trail, because precision matters more than speed in that habitat. Other times, as with mass recruiting ants, the perfume freeway is the right tool and the landscape lights up with invisible signage. Humans borrow the idea in gentle ways. Software inspired by these tricks, often called ant colony optimization, sends many little digital walkers through a problem, lets them lay virtual scent on promising steps, evaporates that scent over time, and watches a path emerge that schedules machines more efficiently. routes data packets more robustly, or arranges delivery trucks so a city breathes easier. The code works not by brute force, but by letting many partial attempts pull each other into a pattern, while forgetfulness keeps the group from worshipping a mediocre first find. And because there is no single controller, the system can tolerate failure and surprise better than brittle top-down plans. If you're tempted to translate this into your own day, do it carefully but concretely. Scatter small scouts into different tasks without overthinking. Leave yourself environmental cues that make good choices easier to repeat. Notes, layout. Friction removed where it helps, and allow your trails to fade so you can pivot when conditions change. Watch how a small timing advantage multiplies when you reinforce it, and how deliberate forgetting keeps you from clinging to what no longer feeds you. And remember that elegance doesn't always look like abstraction. It can look like thousands of feet, each with almost no knowledge, weaving a map that adapts in real time. By the time the kitchen floor is clean and the sugar trail is gone, the wonder remains. A colony that never learned geometry still drew a near-optimal line, not through genius but through simple signals, patient testing, and the humility to erase what isn't working. And there's a soft comfort in that lesson as you close your eyes tonight. The shortest path in a complicated life often appears when you let small, honest actions leave traces. Trust the feedback they generate, and give the future permission to rewrite the route without asking your pride. B. Waggle code. Coordinates danced in the dark. What if a map could be danced where no one can see it? Drawn in the dark on a vertical wall, read by touch and sound rather than by sight. Would you believe it could guide hundreds of travelers to the very patch of flowers you just discovered? Inside the hive, that is exactly what happens as a returning forager steps onto the comb and performs the waggle dance. A figure eight whose straight waggle run carries layered information. Its angle relative to gravity encodes the compass direction of the food relative to the sun outside. Its duration conveys the distance, and its energy hints at the quality. So you are watching a living coordinate system with no ink, and the more carefully you look, the more precise it becomes. The room is almost black to us, but it is alive to them. The dancer vibrates her abdomen while running straight for a second or two, buzzing at a particular pitch, then loops back left or right and repeats. Followers crowd close, pressing their antennae against her thorax and sides to feel the vibrations and micro airflows, tracking the angle of that straight run against vertical, so they read the azimuth to fly once they step into daylight. And in that tiny angle lies a moving sun compass. Because the sun shifts about 15 degrees each hour, the dancer corrects for time using her internal clock, so a run tilted, say, 20 degrees to the right of up now will be tilted a little more later for the same field, and recruits that leave after decoding will head in the correct direction, even though the light outside has changed. Distance is not measured in meters, but in effort, and bees estimate that effort through optic flow, the stream of visual motion across their eyes as they fly. So a path over cluttered fields feels longer than a path over smooth water. You can watch this illusion in controlled experiments where bees trained to fly through a patterned tunnel later dance as if the food were much farther than it really is, because the fast-moving stripes on the walls made the world appear to race past, stretching the odometer in the brain. And the dance faithfully transmits that perception, rather than an absolute yardage. On overcast days, when you might think a sun compass would fail, bees still orient because they sense the sky's polarized light pattern through specialized photoreceptors near the top of the eye, extracting the hidden geometry of daylight even behind clouds. And when the source is very close to the hive, typically within a few dozen meters, the mapping changes into a round dance that drops the direction and simply announces nearby good stuff, trusting recruits to search by scent once they arrive. That scent is not an afterthought, but a key, because the dancer often shares a droplet of nectar with followers, letting taste and smell tell them exactly which bloom to search for when they hit the general area. A clever two-channel code, where the dance gives coordinates and the flavor gives the target's signature. None of this is a legend told to make insects seem human. It is grounded in decades of careful work beginning with Carl von Frisch, who showed that shifting the sun's apparent position with mirrors or moving the hive changed dance angles predictably, and later studies added layers you can almost feel if you stand near an observation hive. The comb acts like a resonant floor, transmitting faint vibrations. The air carries tiny puffs from wing beats, and the crowd noise rises and falls as recruitment gains momentum. When the patch is rich, dances are frequent and persistent. When it wanes, the message dies out, a self-tuning broadcast that matches the economics of the outside world. The hive also shares warnings through other signals most people never hear about. A stop signal, a quick buzz, coupled with a headbutt, delivered to a dancer, can suppress recruitment to a site that proved dangerous, like a feeder where predators lurk or a flower patch sprayed with a repellent. A tremble dance can recruit more receiver bees to process nectar when foragers are returning faster than the bottleneck at the storage area can handle. And a shaking signal can energize resting workers when the colony needs more activity. So the choreography is larger than one message and acts like traffic control across the whole city. Precision has its limits, and those limits are instructive rather than disappointing. The dance rarely points to a single bloom, but to a sector in the landscape. A wedge out from the hive where recruits fan out, and then key on the correct odor to lock in. And the system relies on many parallel attempts instead of perfect individual accuracy. The magic is statistical. Enough recruits land near the target and stick, so nectar flows in without any single bee having to be flawless. And if wind, terrain, or obstacles make the advertised bearing unworkable, flexible pilots adjust en route, and the most successful trips inspire more dancing, while weak ones fade. The code is not the same everywhere either. Different honeybee subspecies show dialects in how dance duration maps to distance, likely tuned by local landscapes. And some use more open-air dances near nest entrances, while others keep it mostly inside. The shared principle stays constant, but the calibration slides a bit as evolution and ecology nudge the curve, reminding you that communication systems adapt to place. When a colony is house hunting during a swarm, the same dance becomes a ballot for real estate. Scouts that find a spacious, dry cavity dance longer and with more vigor, attracting more scouts to check it out. And those newcomers, if impressed, return and dance too, while scouts for lesser sites dance weakly or stop. Cross-inhibition signals help prevent deadlocks as supporters of one site briefly interrupt dances for competitors. And once a threshold of bees gathers at the winning cavity, the swarm warms up and flies as a unit. Destination chosen by a slow crescendo rather than by a boss. Another use of the dance that turns movement into consensus without a committee. For your mind, this is not a cute story about smart insects. It's a lesson in how reliable guidance can emerge from honest, compressible signals coupled to good sensors and a willingness to search. Direction by angle, distance by duration, quality by energy, identity by taste and odor, and safety by brief, corrective shoves. All decoded by followers who do not need to know the theory, only how to align their bodies to a trembling line in the dark. And if you step back from the comb after a few minutes, you feel the design settle in you. A communication system built to work under noise and changing light, built to update as the sky moves, built to forgive error by letting recruits refine the last meters with smell, built to shut itself down when the truth turns, and built to carry a community toward food, with nothing more than a dance you could fit on a postage stamp. It leaves you with a soft question as you drift towards sleep. In your own life, which messages carry enough of the world's structure that others can act without waiting for you to explain everything? And how might you make your signals about direction, effort, quality, and risk just clear enough to guide without pretending to be perfect? The way a bee's map, danced in the dark, still fills the meadow with motion. Neural microcolumns, repeating circuits that think. What if your thoughts were not a single grand computation, but a mosaic of tiny repeating neighborhoods, each just a few hair widths across, stacked in layers, and wired vertically like a bundle of elevator shafts? Because that is one of the most striking ideas about your neocortex. Much of it is built from microcolumns, slim vertical circuits that repeat across the sheet of cortex and give rise to perception, movement, memory, and imagination through shared rules rather than bespoke designs. You can picture a microcolumn as a narrow cylinder that spans the cortical thickness, with most of its neurons being excitatory pyramidal cells whose long dendrites point toward the surface, wrapped by a mesh of diverse inhibitory interneurons that set timing and gain, all organized into 6 layers that have distinct roles. Near the middle, input arrives from the thalamus into a band often called layer 4. Above it, layers 2 and 3 talk to neighboring columns and send feed-forward messages to the next cortical stage. Below, large neurons in layer 5 project out to the spinal cord and deep brain structures to shape behavior, while layer 6 reaches back to the thalamus to modulate the very stream of input coming in. And this laminar choreography repeats across sensory, motor, and association areas with local variations, forming what many neuroscientists have called a canonical microcircuit, not identical, everywhere but recognizable the way a city's blocks can share a familiar grid while hosting very different lives inside. Development gives this pattern a physical logic. During growth, neurons migrate along radial scaffolds from the inner germinal zones outward, settling in waves that create layers, and the axons and dendrites that wire them up show a strong bias to connect vertically within these narrow domains. That bias gives rise to microcolumns, thin stacks sometimes described as tens of micrometers wide, often grouped into larger macrocolumns, hundreds of micrometers across, each macrocolumn containing many microcolumns like streets within a district. In primary visual cortex of primates, you can see this structure mapped onto function as orientation columns that prefer edges tilted at specific angles, arranged in smooth pinwheels so neighboring columns like similar orientations. And layered on top of that are eye of origin stripes, called ocular dominance columns, that tile the same space so that each local patch of cortex holds, in miniature, a balanced sample of the visual world. In rodent somatosensory cortex, whisker inputs are represented in barrels, column-like modules that keep signals from individual facial whiskers organized so precisely that poking one hair activates its matching barrel. A vivid demonstration that vertical circuitry is not a cartoon, but a working address system. If you zoom into the microcolumns wiring, you find motifs that feel like basic grammar. Small clusters of pyramidal neurons excite one another and form recurrent loops that can amplify faint inputs into clear signals. Fast-spiking inhibitory cells curb runaway excitation and sharpen timing so that bursts become crisp, not mushy. Other inhibitory types reach across layers or wrap the dendrites of pyramidal cells to gate plasticity, deciding when learning is permitted. Neuromodulators like acetylcholine and noradrenaline slide the operating point of the circuit, opening or closing the aperture so the column becomes more exploratory or more selective depending on state. And all the while, rhythms emerge, brief ripples of synchronized activity in faster bands during local processing, slower waves that carry feedback and context, so that columns can talk within neighborhoods and across distant lobes without shouting over each other. The functional promise of all this repetition is not monotony, but reusable computation. The same template that detects an oriented edge in one place can, with different long-range inputs and training history, help encode a spoken syllable, a remembered face, or a planned grip. Learning then looks like retuning a shared device rather than wiring an entirely new machine, with synapses strengthening and weakening inside and between columns, as experience sculpts the flows that matter to you. There is real debate here, and it keeps the story honest. Not every species shows crisp columns in every area. Some functions spill across columns more than early theories suggested, and the edges of a column are fuzzy rather than hard walls. Yet the vertical bias of connections, the laminar division of labor, and the recurrence of similar motifs across vast territories are robust findings, and they support a helpful working picture that cortex scales by copying a flexible microcircuit and then weaving columns together in different patterns. Thinking about computation in this way nudges you to a practical insight. When you hold a scene steady in mind, you are stabilizing local recurrent loops so they sustain a pattern for a moment. When you suddenly recognize a face in a crowd, a sparse coalition of columns primed by past learning locks into a configuration that fits the input better than the alternatives. When you predict what comes next in a melody or a sentence, higher areas send feedback down columns in earlier areas to bias what they will accept as a good match. And mismatches create small bursts of error that adjust the current hypothesis. Nothing mystical, just layered circuits passing proposals up and constraints down until the network settles. Because the blueprint repeats, damage can be both devastating and surprisingly specific. Disrupt columns in a particular patch of visual cortex, and you lose a narrow slice of the map, a scotoma. Disrupt a set in the motor strip, and certain movements falter while others remain intact. Even in disease, column-level patterns emerge. Some conditions show altered spacing or inhibitory balance at the mini-column scale, which researchers explore not as slogans, but as clues to how micro-level miswirings ripple into perception and behavior. Tools have made this landscape vivid. Microelectrodes can record from many neurons across layers in a single column, while imaging shows how activity spreads like ink through stacked sheets. Optogenetics lets scientists nudge one cell type at a time, turning on fast spiking interneurons to quiet a loop or silencing layer 4 to test what happens when the thalamic gate closes. And the results, while nuanced, keep pointing back to the same refrain. Local rules plus long-range context create meaning. Even your sense of time and confidence in a decision seem to emerge from how columns pass and hold activity, with some regions integrating evidence over seconds and others resetting quickly for the next frame, so that the mind's smooth stream is assembled from many local pulses agreeing just long enough to act. Step back now and the conclusion feels both humbling and empowering. Your neocortex is not a bespoke palace, but a cathedral built from repeating arches, each arch small enough to fit under a grain of rice, each arch capable of a handful of elemental operations: match, amplify, inhibit, compare, predict. And from their endless tiling and flexible conversation comes a life of color, language, and choice. The beauty is that this design offers resilience and learning without requiring an overseer. So you can trust that small local changes, attention here, practice there, really can rewire what you perceive and how you decide. As you let that settle, the thought grows gentle. Inside your skull, millions of tiny neighborhoods hum in parallel, each doing its part with the same quiet recipe. And from that chorus of repeating circuits, your memories, your habits, your hopes, emerge not as a miracle of complexity for its own sake, but as the natural song of a pattern, simple enough to repeat and rich enough to think. Astrocyte networks, calcium waves that carry messages. You've been taught to hear the brain as a crackling forest of electrical spikes, but there is a quieter language layered underneath, slower, softer, and spread through star-shaped cells that do not fire like neurons yet still carry meaning. Astrocytes form sprawling networks that lace your synapses and blood vessels, and they talk in shimmering calcium waves that rise and fall like moonlit tides. To meet them properly, imagine a single astrocyte as a many-armed caretaker whose fine processes wrap thousands of synapses while its end feet hug nearby capillaries. So one cell touches the places where information is exchanged and the pipes that deliver the fuel. And when neurotransmitters spill into the synaptic space, glutamate from an excited neuron, for instance, receptors on those astrocytic processes respond not with a spike, but with a ripple of calcium inside the cell. A chemical pulse that can stay local in tiny microdomains or spread through the entire cell and even jump to neighbors, turning a cluster of glia into a living field of slow signals. The mechanism is earthly, not magical. Many astrocytes carry metabotropic receptors that, when activated, trigger internal stores to release calcium. And because astrocytes are linked by gap junctions made of connexin proteins, they share small molecules and pass news along, while ATP released into the space between cells can act as a messenger that binds to purinergic receptors on nearby astrocytes, creating a relay that travels across tissue for hundreds of micrometers in seconds, a wave without wires. What those waves do is easier to grasp when you stop thinking of astrocytes as mere support and see them as governors of context. At synapses, they vacuum excess glutamate through transporters, preventing toxic buildup and sharpening neural messages. They help mop up potassium when bursts of neural activity push ions out of balance, keeping excitation from spiraling out of control. And they release small molecules, sometimes called gliotransmitters, like ATP or D-serine, that can tune how receptive a synapse is. Though here the literature is careful. In reduced preparations, astrocytes plainly influence synaptic strength, while in living brains the size and timing of that influence vary with state, region, and method, so the field keeps testing rather than preaching. Their reach extends from circuits to circulation. Those end feet around blood vessels are positioned to regulate flow, and calcium rises in astrocytes can lead to the local release of vasoactive substances that dilate or, in some cases, constrict nearby arterioles, redirecting blood toward patches of working tissue. The practical consequence touches you every time you see an fMRI image, because the bold signal that maps brain activity depends on changes in blood oxygenation that astrocyte-guided neurovascular coupling helps produce. So your picture of thought owes a debt to cells that manage plumbing as part of computation. Zoom in further, and the quiet language becomes nuanced. Astrocytic calcium is not a single kind of event, but a menagerie of flashes, brief, micrometer-scale sparkles in fine processes, broader waves that sweep through a branch, and, under strong drive, cell-spanning surges that enlist neighbors. Different neuromodulators, like noradrenaline or acetylcholine, bathe networks during arousal or attention and reshape these patterns, priming astrocytes to respond more readily or more selectively, so the same sensory input lands in a different internal weather. In the suprachiasmatic nucleus, the brain's clock, astrocytes display their own daily rhythms and can influence neuronal timing, hinting that your circadian tempo is co-authored by cells that rarely get marquee billing. At night, their role deepens in a way you can almost feel when you wake clearer after good sleep. Astrocytes help build the signal for sleep pressure through the slow accumulation and release of adenosine, which makes neurons less likely to fire. During deep sleep, water channels clustered in astrocyte end feet support convective flow through brain tissue, the so-called glymphatic system, that washes out metabolic waste more effectively than during wakefulness, a housekeeping surge that may help explain why disrupted sleep correlates with the buildup of protein aggregates in disease, though cause and effect are still being untangled. In development and plasticity, astrocytes act as quiet project managers. They secrete molecules such as thrombospondins and heavin that encourage synapse formation, and they can also prune connections by engulfing weak synaptic elements through dedicated receptors, helping sculpt circuits so that what remains is cleaner and more efficient. When learning happens, neurons change their synapses, but astrocytes change their coverage and transporter levels, altering how vigorously a synapse is buffered, which in turn shapes the probability that a pattern of activity will stick. And this layered control means memory is not only in the wire, but also in the room around the wire. Inevitably, the system's strengths trace the outline of its vulnerabilities. When potassium buffering falters or glutamate uptake lags, networks can drift toward hyperexcitability, contributing to seizures. When astrocyte channels or junctions are mislocalized or altered, blood flow responses can become sloppy. When calcium signaling runs too hot or too cold, inflammatory cascades or metabolic mismatches can follow. And in conditions from migraine aura to neurodegeneration, researchers keep finding astrocytic changes that look less like passive damage and more like active participants in a complex pathology. Signals, however, are not guilt, and separating cause from consequence remains a careful, ongoing task. If you enjoy scientific debate, astrocytes offer a healthy one. Early models emphasized broad, IP3-driven calcium waves as master regulators. Newer in vivo imaging reveals many fast, local microdomain events, even in mice lacking a major IP3 receptor subtype, suggesting multiple pathways feed the same language, and that context decides which dialect is spoken. Likewise, gliotransmission has moved from bold general claims to nuanced, cell-type and state-specific effects, with many labs converging on a middle view that astrocytes do modulate synapses and networks, but often through subtler, time-integrating mechanisms that complement rather than mimic neuronal spikes. The most practical way to appreciate their intelligence is to follow energy. Astrocytes sit at the crossroads of fuel supply and demand, storing glycogen, shuttling lactate to neurons that are burning fast, and timing blood flow tweaks so that the next volley of spikes has oxygen and glucose on tap. In this sense, calcium waves are not idle glows but dispatch orders. Where to tighten cleanup, where to ease vessels open, where to lift or lower the gain on local circuits, so the support cells are, in truth, managers of readiness, ensuring that brief electrical thoughts can arise against a stable, nourished background. Let the conclusion settle softly. Beneath the sharp chatter of neurons, your brain keeps a second conversation in murmurs of calcium that drift through a mesh of glia, coordinating supply with demand, pacing rest with activity, and adjusting the space in which signals make sense. Learning this does not require you to romanticize or to pick sides in old arguments, only to widen your picture of what thinking involves. Because part of your clarity tonight, your ability to follow a line of ideas without fatigue, depends on cells that never announce themselves, carrying messages in waves you can't feel, keeping the house in order so the mind upstairs can stay lit. Immune swarms, distributed intelligence, without a brain. You nick your skin on a sharp edge, and before your mind can form the thought injury, a plan assembles itself from cells that have never seen a blueprint. First responding sentinels at the wound release urgent chemical beacons that spread through tissue. Nearby blood vessels loosen their grip so help can slip out. And the body's fastest foot soldiers, neutrophils, home in. Not by following a single instruction, but by reading overlapping hints in the air. They sense tiny gradients of alarm molecules and, as each one arrives, it emits its own amplifiers, especially a lipid called leukotriene B4, that strengthen the scent for others. So what begins as a scattered trickle becomes a coordinated rush in minutes, a living surge that circles the threat, contains it, and then, if all goes well, fades. This is immune swarming. Distributed decision-making written in receptors and relays instead of words, where pioneer neutrophils that first touch danger ramp up the call, second waves reinforce the signal, and latecomers spread around the perimeter, creating rings of containment that you can watch under a microscope as rippling constellations of white cells converging and stabilizing. Each cell runs on simple rules. Move up the steepest rise of specific chemicals. Pause when receptors saturate. Release boosters when contact is made. Yet the swarm as a whole shows strategy, forming a cordon where pathogens are most likely to escape while leaving routes open for repair. The beacons themselves form a hierarchy. Fragments from damaged microbes and tissue act as long-range lures, while locally produced signals, including LTB4 and various chemokines, focus the final meters. Cells adapt as they climb, temporarily desensitizing some receptors so they don't stall in weak fields, and switching to fresh cues near the core, which keeps traffic flowing rather than clogging at the first hint of distress. Once at the scene, neutrophils change shape and behavior. Some engulf invaders. Others spill sticky webs of DNA and proteins, extracellular traps, that snare and neutralize threats too large or slippery to swallow. And many simply die after a short tour. Their corpses offered up to macrophages whose quiet consumption of the fallen is more than cleanup. It is a biochemical vote to de-escalate, because eating apoptotic cells flips a switch in these janitors toward releasing calming molecules that reduce recruitment and begin the handoff to healing. This turn toward resolution is as active as the attack. Specialized lipids like lipoxins and resolvins rise. The tissue's chemistry tilts from "come here now" to "stand down." Fibroblasts and endothelial cells repair the scaffolding. And the same network that magnified alarm learns to quiet itself, because a swarm that never stops becomes damage without purpose. The difference between life-saving inflammation and a dangerous storm is not raw power, but feedback. Enough to rally, enough to restrain. The immune system's distributed intelligence is not confined to battlefields in skin. Inside your lymph nodes, rare decision-makers hold elections every day without a chairperson. Dendritic cells roam tissues sampling events, then carry carefully packaged fragments of what they saw to the node, where they park at junctions and present those fragments to passing T cells. The odds are terrible. Only a handful of T cells in the entire body may fit a particular fragment, but traffic solves it, because thousands of T cells drift along defined corridors and scan one presenter after another, lingering only when their receptors catch a match. If the match is strong and the context screams danger, the T cell shifts state, begins dividing, and adopts a role: helper, killer, regulator, guided by the mix of cytokines and signals around it. Within days, clones flood the network, each with the same specific recognition, a distributed army grown on demand from a single lucky find. In nearby follicles, B cells run a different, yet equally emergent program. They test their receptors against the same fragments, seek help from specialized T cells, and, if chosen, dive into germinal centers, two-zone workshops where they rapidly mutate their receptor genes, then compete for survival by binding targets presented on follicular dendritic cells. Those that improve are sent back to proliferate again. Those that falter exit, and over cycles, the population sharpens into cells that produce antibodies with better and better fit. No authority designs these tournaments. Selection pressure, space, and time do. Elsewhere, other swarms patrol with their own styles. Natural killer cells sweep tissues in loose lattices, pausing just long enough to interrogate suspicious cells and withdrawing if a self badge is shown. Interferon released by infected cells turns neighborhoods into antiviral states, an early warning broadcast that buys time for slower adaptive forces to arrive. Tissue-resident memory cells, once formed after an infection or vaccine, take up long-term posts at likely entry points and answer local alarms within minutes, a distributed picket that prevents small sparks from spreading. None of this works without breaks, because enthusiasm can kill as surely as negligence. Regulatory T cells damp excessive T cell expansion. Macrophages that have eaten enough of yesterday's chaos become anti-inflammatory, and soluble decoys soak up chemokines so trails do not sharpen forever. When these checks falter, you see the cost in autoimmune flares, chronic inflammatory diseases, or runaway cytokine cascades, failures of governance in a polity that normally polices itself. The elegance here is that useful complexity arises from cheap parts. A GPCR that smells a gradient, an adhesion molecule that lets a cell squeeze through a vessel wall, an enzyme that spins a lipid into a louder call, a receptor that recognizes a fragment by shape, and from their interplay comes recognition, response, memory, and mercy. None of it stored in a single place, all of it stored in the habits of a crowd. You can test the system's intelligence by challenging it, and medicine does. Vaccines do not push a button so much as present a safe rehearsal that seeds memory B cells and T cells across the network, so that the next time a similar threat shows up, the swarm remembers the more efficient playbook. Modern therapies sometimes succeed not by poisoning enemies, but by modulating the conversation, blocking one cytokine to break a feedback loop in arthritis, releasing an inhibitory checkpoint on exhausted T cells so they re-engage tumor antigens, or supplying resolvin-like molecules to help a system stuck in attack find its way to detente. If you listen with the right kind of attention, the conclusion is quiet but profound. You are protected by a society of cells that coordinate without a headquarters, that decide by local rules and global consequences, that recruit, escalate, and then relinquish control so tissues can return to ordinary life. And the question this raises is gentle and useful. When you face problems that feel too large for a single mind, can you build processes that act like this, clear about when to gather, when to amplify, and when to let go? Because in your bloodstream tonight, legions that never learned a language still manage to keep the peace, and their wisdom is not a metaphor, it is your pulse. Cellular automata in tissue, rules that sculpt bodies. Trace the back of your hand with your eyes and ask a simple, unsettling question. How did a formless cluster of cells decide where to place every ridge, follicle, vessel, and tendon with such quiet confidence? Because bodies aren't sculpted by a foreman shouting orders. They are carved by local rules repeating everywhere, the biological equivalent of a cellular automaton in which each cell updates its behavior by reading its neighbors. And from that humble conversation stripes, segments, branches, and folds emerge on schedule. In development, a cell doesn't see the whole plan. It samples short-range chemicals, tugs and pushes, and tiny differences in concentration or tension become if-then instructions. Divide now, pause, migrate, stick harder, soften and fold. One classic rulebook is called lateral inhibition. When two adjacent cells both want a special fate, a notch delta handshake between them lets the slightly stronger one suppress its neighbor, spacing out winners into a checkerboard of sensory bristles in a fly or hair cells in your inner ear so they don't clump. No map is drawn, yet precision appears because each pair resolves its own tie. Another is the activator-inhibitor duet that Alan Turing imagined, long before biologists could watch it. One molecule that locally encourages a fate while helping produce a diffusing break that spreads farther and damps it at a distance. The push-pull can seed regularly spaced follicles on skin, palate ridges in the mouth, or the spacing of buds that will become fingers. And although the specific molecules differ by organ, the logic repeats like a rhythm section you recognize in different songs. Some patterns aren't laid by chemicals alone, but by oscillations in time. Along the embryo's back, cells in the pre-Semitic mesoderm tick in genetic rhythms, waves of gene activity that sweep from head to tail, while a biochemical wave front moves slowly in the opposite direction. Where the wave and the front meet, a segment freezes into a somite, the seed of vertebrae and ribs, and the metronome is so disciplined that a defect in the clock's gears can lead to scoliosis or extra ribs, proof that timing is as much a rule as position. Direction itself can be a local decision. In planar cell polarity, small complexes on the surfaces of neighboring epithelial cells bias one another, aligning hairs on your forearm or the orientation of cilia in your airways, so that a tissue gains a shared arrow without consulting a compass. The resulting coordination is vivid when it fails, as cilia beat out of tune and mucus clearance stumbles. Patterns also arise from mechanics that any child's hands can feel. If a sheet of cells constricts at its top surfaces more than at its base, it buckles inward to form a furrow, the first gesture of a neural tube or a gut. And if particular cells soften or stiffen relative to their neighbors, ridges and villi sprout like a neatly plowed field. In lab dishes and embryos, apical constriction, differential adhesion, and tissue tension repeatedly generate folds and tubes with no one drawing lines. And the rules here are not mysterious. Each cell acts on the forces and attachments it senses, and the sheet as a whole settles into shapes that balance those forces. Cell sorting is another automaton-like rule that plays out with startling inevitability. Put mixed, dissociated embryonic tissues together, and they often reassemble by adhesion strength, with stickier types sinking to the center and looser ones forming the outside. A self-organization that helps re-establish compartments after injury, computational models that assign each simulated cell a preference for certain neighbors reproduce this sorting, and even predict how sharp a boundary will be, giving engineers a way to test designs for artificial tissues. Branching organs follow their own neighborhood laws. In developing lungs and kidneys, tips that are growing avoid crowding other tips, split when they reach a certain size, and slow down if local inhibitors accumulate. So a few rules: grow, bifurcate, respect personal space, yield tree-like airways and ducts that fill the available volume efficiently, tweak the thresholds and you change lobes and duct counts, underscoring how small parameter shifts in local behavior scale to visible anatomical diversity across species. Even skin color patterns can be read as living automata. In zebrafish, stripes aren't painted by a distant brush. They arise from pigment cell types that attract, repel, and transform each other at short range. Cells extend filigree processes to feel neighbors, keep distance from rivals, and recruit allies. And when a patch is injured, the conversation regrows the missing bars with fidelity, revealing that the rules are stored in the interactions, not in a static template. If you fast forward to today's tools, you see scientists both watching and editing these rules directly. Light sheet microscopes capture millions of cells in embryos as they divide and fold, turning guesswork into time-lapse evidence. Optogenetic switches let researchers turn a signaling pathway on in a precise patch for minutes and watch a new stripe appear on command. Synthetic biologists have built simple, synthetic notch circuits in mammalian cells that recreate lateral inhibition or even produce designer spots and borders, demonstrating that a handful of parts and local coupling can write a pattern, which is thrilling because it hints that future therapies might one day repair a malformed tissue by nudging rules rather than replacing parts. None of this requires equations on your pillow, only the humility to see how global form can be a side effect of local sense. Cells compete, cooperate, and compromise with neighbors using short messages and gentle pushes. The settlement they reach at every square millimeter becomes shape. And when the rules wobble, you can read the consequences. A digit that never splits cleanly leaves webbing. A clock that runs fast adds an extra vertebra. A polarity map that blurs makes cilia point the wrong way, and chronic infections follow. Equally, when rules restart after injury, remarkable things can happen. Liver regenerates with proper lobules. Intestinal crypts rebuild their repeating architecture. Organoids grown from stem cells self-organize into structures with buds and layers that look eerily like their full-size counterparts. All because the update program is carried by each participant. Tonight's gentle takeaway is that your body's reliability is not a miracle of centralized control, but a chorus of tiny agreements. If this neighbor is of type A, become type B. If the local inhibitor is high, pause. If tension rises here, fold there. If a clock strikes, freeze the segment. You can feel the philosophical warmth of that idea without forcing it. Complex beauty doesn't demand a mastermind. It emerges when small actors follow honest rules and keep listening. And if you wonder what to do with such knowledge in your own life, you might start by tending to the neighborhoods of your habits and relationships, the short-range cues you send and read each day, because large changes often appear, not when you redraw the whole map, but when you refine the little rules by which adjacent moments update one another until a pattern you can live in takes shape. Developmental gradients, morphogen maps that draw you. Before you had a face, before you had a spine or hands or even a left and a right, Your body was a field of quiet slopes, chemical hills and valleys that told each cell where it was and what it should become. And those invisible contours, called developmental gradients, are the maps that drew you. In the simplest picture, a small group of source cells produces a signaling molecule, a morphogen, that diffuses outward and is soaked up, degraded, or carried away, so its concentration falls with distance. Cells sample that concentration and, like hikers reading altitude, choose different genetic programs at different levels. But the reality is more beautiful than a static fog. Morphogens don't just waft aimlessly, they bind to the matrix between cells, ride along tiny cellular projections, get internalized by receptors and passed hand to hand, and are shaped by tissues that are themselves moving and growing. So the gradient is a living river rather than a painted stripe. Early in a fruit fly embryo, for example, a protein called bicoid gathers highest at what will become the head and trails off toward the tail. Nuclei bathing in high bicoid switch on one set of genes. Those in middling levels choose another, and those in low levels choose a third. This is the heart of the positional information idea. A single chemical slope can carve the field into crisp bands by setting thresholds, and each band then builds finer detail. In vertebrate embryos, a different slope of sonic hedgehog streams outward from a thin midline structure to pattern the future spinal cord. Cells closest to the source become floor plate and motor neuron precursors. Cells a little farther out adopt other neural identities, and the borders sharpen because neighboring gene networks push each other apart. Turn on one fate, and it actively suppresses the alternatives, so a fuzzy concentration becomes a clean boundary. In a developing limb bud, Sh again acts at the back edge to instruct digit identity, a fact you can feel in a classic experiment. Implant a little bead soaked in Sh into a chick limb and extra, mirror image digits sprout, a demonstration so striking that it's hard to forget once you've seen the photographs. Other gradients layer on top. BMPs and their antagonists set the back versus belly architecture. Eints and retinoic acid shape axes and segment identities. Nodal signals help establish left-right asymmetry, so your heart bends the correct way. Cells aren't naive readers, though. They don't just glance at concentration and decide. They integrate over time, measure the steepness of the slope, and listen to the duration of exposure. In the spinal cord, for instance, Sh does double duty. High levels tilt gene regulators strongly, but long exposure deepens the commitment, and the combination of level and time creates nested territories of identity that are far more robust than a single snapshot could be. This temporal hearing matters because embryos are busy places. Sources move, tissues expand, and molecules fluctuate. By averaging in time, cells can ignore flicker and commit only when a signal is persistently convincing. You might wonder how such a system avoids breaking when embryos vary in size. Yet gradients often scale. If the field is bigger, sources adjust output and sinks adjust uptake so that the pattern stretches proportionally, keeping stripes and territories in relative position. Some organisms achieve this with feedback, morphogens, induce the production of their own inhibitors or receptors, so the shape of the curve self-corrects, while others rely on multiple sources that define ratios rather than absolutes. So cells decide based on fold changes from baseline rather than raw counts. The organizer experiments that change developmental biology forever show how powerful a shaped field can be. In the 1920s, grafting a small cluster of cells from one side of a salamander embryo to the other didn't merely add tissue. It sent signals that reoriented neighbors and built a second, well-formed axis. Those organizer cells secreted antagonists that sculpted BMP gradients and invited a new head-tail and backbelly structure to form from ordinary surrounding tissue. The lesson isn't that a magical master cell holds the blueprint, but that a properly positioned source can reshape the entire conversation, and cells will follow the map if the signals are honest and sustained. Precision arises from the way gene networks read these cues. Many developmental genes carry enhancers, short stretches of DNA, that respond only when a specific combination of morphogen-activated factors binds at once. Below a threshold, the enhancer remains quiet. Just above it, the enhancer switches on sharply, and slightly different enhancers with slightly different thresholds engrave multiple bands from a single slope. Add cross-repression, where each chosen fate actively discourages its neighbors in the same cell, and intercellular no trespassing around borders, and you get crisp stripes that don't bleed when growth or noise jostles them. This is how a vague gradient becomes a precise map. Layers of thresholds, mutual exclusivity, and neighborhood policing turn softness into structure. Transport is more than diffusion, too. In some tissues, specialized filaments called cytonemes extend from one cell to another, delivering morphogens like hand-addressed letters so that target regions see a tailored dosage. In others, morphogens bind heparan sulfate chains in the extracellular space, briefly immobilizing and then re-releasing them, a stop-and-go that limits overshoot and keeps profiles steep. Receptors themselves shape the field. By grabbing molecules at the surface and pulling them inside for degradation, they act as mobile sinks that sharpen the gradient where receptor levels are high. This receptor-mediated clearance creates a steeper drop-off than diffusion alone could achieve, especially near hungry borders. If you step back, the reliability of gradients comes from redundancy. Rarely does one molecule rule an axis. Instead, overlapping families, BMPs with their antagonists, W and TES with modulators, Hedgehog with its feedback, co-author the map so that if one stream runs a little thin, the others fill in the slope. And when something does go wrong, the phenotypes reveal the underlying coordinates. Damper limbs shush signal and digit identities shift from back toward front. Ramp it up or hold it too long, and extra posterior digits appear. Disrupt retinoic acid processing and the address labels on your vertebral column, the Hox code, slide, transforming one vertebra into the identity of its neighbor. Tweak the gradient reading machinery, receptors, intracellular transducers, or the enhancers that set thresholds, and borders blur. Tissues misplace themselves, or symmetric structures acquire asymmetry where none belongs. These aren't curiosities. They're how physicians and researchers trace congenital disorders back to missed steps in a once silent map. Modern tools let us watch and even repaint gradients in living tissue. Fluorescently tagged morphogens reveal moving fronts and stable plateaus. Microfluidic devices bathe cells in controlled slopes to test how thresholds and timing interact. Optogenetic switches turn light into a proxy morphogen, allowing scientists to sculpt synthetic gradients across an embryo and see fate maps redraw almost instantly. And in organoids, mini-organs grown from stem cells, engineers pattern sources and sinks around tiny spheres to coax layered retina, ordered brain regions, or gut tubes to self-organize. Proof that when the map is present, matter follows. The deepest comfort here is philosophical but grounded. No master architect had to touch every place at once. Local measurements of a common field, combined with simple, enforceable rules inside each cell, were enough to create a coherent whole. A gradient doesn't tell a cell be a finger. It says, you are this far from the source and that far from the edge. At this level and time, turn on these switches, and millions of such modest instructions compose a hand. As you let that sink in, it reshapes how you think about complexity in your own life. When the big picture feels overwhelming, useful maps can still be drawn from a few honest signals if you choose thresholds wisely, read them consistently over time, and let boundaries sharpen where they should. The science does not need embellishment. Gradients exist, cells read them, and from those sloping whispers your body took its shape. Direction without a director. A map that drew you while you slept in the smallest darkness. Gene regulatory circuits, switches that decide cell fate. Suppose I told you that the difference between a beating heart cell and a thought-bearing neuron isn't a new set of genes, but a new way of reading the same book. That inside each cell, tiny decision circuits flip molecular switches that choose which chapters to perform and which to keep closed. And once you see that, development and healing start to look less like magic and more like careful governance under noise. A gene regulatory circuit is built from transcription factors that latch onto short stretches of DNA near genes, enhancers, and promoters, plus the chromatin machinery that opens or tightens the local packaging so those factors can work. When a factor turns on a gene that returns to strengthen the same factor, you get positive feedback that can lock a choice in place. And when two factors turn each other off, you get a tug of war that forces a winner, the molecular version of a vote that doesn't end in a tie. Cells use these motifs to turn gradual hints into decisive outcomes. A dim nudge from the environment might start a trickle of a key factor. That trickle wakes the gene that makes more of it. The loop catches, and suddenly the circuit snaps into a stable on state. Because the loop feeds itself, the state can persist after the outside hint fades, which is why a committed muscle cell keeps being a muscle cell when the original instruction is long gone. You can watch this logic in real lineages. In the blood, early precursors face a crossroads. Become a red blood cell lineage or turn toward immune fates. A pair of factors, Garter-type proteins and PU1, push for opposing identities and repress each other, so small early differences, helped by surrounding signals, get amplified until one program takes over and the other cannot easily return. In the embryo's pluripotent core, a triad of factors, OCT4, SOX2, and NANOG, stabilizes the state of being able to become many things, turn the balance and the cell tips into specific fates. And because these factors regulate their own enhancers and those of their partners, the landscape has ridges and valleys rather than a flat plane. Once the cell rolls down into a valley, it resists being pushed back up. That metaphor, often drawn as Waddington's landscape, is not just a pretty picture. It captures the way circuits confer memory, a property you can feel in experiments that reprogram identity. When scientists force just a handful of transcription factors into skin cells, famously a set that includes OCT4, SOX2, CALF4, and C-MIC, those cells climbed out of their committed valleys and settled into a pluripotent valley again, proving that pushing the right switches in the right order can rewrite fate. Other combinations can transdifferentiate one adult cell type into another more directly, like driving fibroblasts into muscle with MyoD or toward neurons with ASCL1, BURN2, and MIT1L. And although efficiencies and safety remain real concerns, the principle is clear. Fate is a circuit state, not a permanent tattoo. Hidden within these decisions is a subtler truth that matters for understanding both robustness and disease. Gene expression is not a smooth faucet, but often a bursty drip, with transcripts made in pulses as polymerases engage and pause and enhancers, briefly recruit large assemblies. To thrive in that noise, circuits use feedback to filter and average. Negative feedback, where a factor or its target suppresses its own production, can steady a signal so it doesn't overshoot. Positive feedback can turn a graded input into a clean yes or no, what biologists call ultrasensitivity, producing a sharp transition even if the upstream cue is merely modest. Some circuits also implement timing logic. A common pattern called a feed-forward loop lets a master factor activate a target directly and also activate a helper that takes time to build. The target will only turn on strongly if the input persists long enough for the helper to arrive. So brief. Noisy flashes are ignored while sustained instructions are obeyed. This is how cells avoid committing to a fate based on a momentary gust of signal in a crowded embryo. The stage on which these actors play is as important as the actors themselves. DNA is wrapped around histone proteins and folded into loops that bring faraway enhancers into polite contact with the promoters they control. Boundaries in this three-dimensional architecture corral interactions, so the wrong enhancer doesn't hijack the wrong gene, and proteins such as CTCF and cohesin help maintain those neighborhoods. When a cell commits, it often lays chemical marks on histones and sometimes on DNA that make chosen regions easier to access and unchosen regions harder. These modifications don't change the letters of genes, but change how readable they are. A form of epigenetic memory that combines with feedback to make identity durable. Some transcription factors are pioneers. They can open stiff chromatin and invite others in, setting the stage for a new program. Others follow later to amplify and refine. In recent years, researchers have also found that clusters of enhancers near key identity genes, so-called super enhancers, can form droplet-like condensates that gather transcription machinery into dense hubs, boosting output in a way that looks less like a single switch and more like a buzzing market square. Perturb one stall and the market shrugs, but disturb the hub and the whole district dims, which may explain both the resilience of normal identities and the fragility of some cancers that lean too hard on one hub. Because these circuits matter so deeply, tools to read and edit them have transformed our understanding. Single-cell RNA sequencing reveals which genes are on in thousands of individual cells at once, drawing branching maps of decisions as they unfold rather than averaging them into a blur. Assays that profile chromatin accessibility show which doors are open as fate changes. CRISPR-based perturbations let scientists nudge specific enhancers and watch how the circuit reroutes, sometimes uncovering cryptic backups and sometimes revealing a single indispensable switch. The lessons are practical. In regenerative medicine, you want to push cells toward healing states without risking runaway growth. Knowing which combinations of factors and enhancers create a safe, stable valley helps design protocols that mature into the right tissue and stay there. In oncology, tumors often hijack developmental circuits, turning on embryonic programs, building new super-enhancers, or mutating boundary elements so genes fall under the sway of inappropriate switches. Drugs that target the epigenetic writers or readers propping up those aberrant states can push malignant cells out of their pathological valleys and back toward more ordinary behavior or susceptibility. In immune therapy, flipping circuits that set T-cell exhaustion or persistence can determine whether a therapy fizzles or endures. Even in ordinary physiology, these ideas explain the grace of long-term change. When you learn a skill, you are not adding a chapter to your genome, but strengthening particular assemblies of factors and access at specific sites, so that the next time your neurons are asked to play that song, the circuit takes the path of least resistance. Honesty keeps this beautiful picture grounded. Not every fate hinges on a tiny number of master switches. Many choices emerge from broad, distributed changes that accumulate across dozens of loci. And cells are not machines waiting for a single lever. Context, metabolism, and mechanical forces from neighbors bias outcomes in ways that pure DNA logic can't entirely capture. Circuits can jam or wobble. Too much noise in early development, or mutations in a pioneer factor, or a misplaced boundary can leave cells stranded between identities, with consequences that echo across a tissue. And reprogrammability is not a guarantee that identities are interchangeable without cost, because developmental time embeds constraints that must be respected for safety. Yet the central insight holds and is quietly liberating. Your body's variety is a performance of shared parts arranged by circuits that read, remember, and decide. When you imagine those decisions as switches that integrate gentle signals, level and duration, partner in place, you start to see fate as a conversation that cells have with themselves over hours and days. A conversation that, once settled, hums on without constant supervision. Let that settle as you close your eyes. There is no single conductor in the orchestra of your tissues, only sections cued by small, local sheets of music. When the entrance is right and the players reinforce one another, a theme takes hold and becomes identity. And the miracle is not that the song begins, it is that, once begun, it knows how to continue. Quorum sensing. Bacteria vote before they act. You don't usually imagine bacteria as a crowd that waits, listens, and then moves together. But picture this. You're a single cell in warm seawater, quietly releasing tiny molecules into the world and counting how many of those same molecules drift back to you. When the count is low, you keep your secrets. When it rises past a threshold, you unlock a different set of genes and act in concert with your neighbors. That is quorum sensing, a density-dependent way of regulating behavior using chemical signals called autoinducers, and it lets microbes switch from me to us with surprising precision. In simple terms, each bacterium produces a signal and also carries a sensor for that signal. If the environment is thinly populated, signals diffuse away and the sensor stays quiet. If the community is thick enough, signals accumulate and trip molecular switches that turn on shared programs, glowing together, building protective biofilms, secreting enzymes, even launching an attack only when it will matter. The best-loved example lives in a nighttime partnership. Vibrio fischeri, a marine bacterium, lights up the belly of the Hawaiian bobtail squid, so the squid's shadow disappears against moonlit water. The bacterium's LuxI enzyme makes an acyl homoserine lactone signal. When enough cells are present in the squid's light organ, that AHL binds a LuxR protein and flips on the genes for bioluminescence. No master brain calls the cue. The community's own chemistry sets the stage, and the timing emerges from a simple rule: produce, detect, respond, repeated by many. Not every species uses the same toolkit. Gram-negative bacteria often rely on AHLs and LuxA-like pairs. Gram-positive bacteria export small peptides and sense them with two-component systems. There's even a bridge signal called AI2 that many species can tune into, a kind of microbial lingua franca that hints at cross-species awareness. You can think of the threshold not as an on-off light, but as a steep dimmer. Once signal curves slope upward, positive feedback makes the change feel sudden and collective. Yet it's all local sensing and ordinary diffusion. Why evolve such choreography? Because some actions only pay off when done together. Enzymes that digest big food molecules, iron-scavenging compounds like siderophores, sticky matrix that cements a biofilm. These are public goods. One cell's contribution is costly and small. 1,000 cells' contribution is transformative. Quorum sensing delays the expense until the return is worthwhile. In pathogens like Pseudomonas aeruginosa, coordinated gene programs controlled by interconnected LAS, RHL, and PQS signaling systems can switch on suites of virulence factors and biofilm formation when the infection is established, explaining why timing matters in disease progression. In Staphylococcus aureus, the Agar system uses peptides to decide between sticking around as a biofilm and dispersing to invade new tissue. Even in friendly soil dwellers such as Bacillus subtilis, peptide signaling can coordinate competence, the temporary ability to take up DNA from the environment, or steer cells towards sporulation when resources run thin. This is not mind or morality, it is chemistry shaping strategy. Yet chemistry alone doesn't guarantee harmony. If producing signals and public goods is costly, why not be a cheater that listens to the quorum and enjoys the benefits without paying? Microbial communities have answers shaped by evolution and physics. Spatial structure keeps relatives close, so helpers mostly help kin. Some lineages couple helpful behaviors with private benefits. Others add policing tactics, linking toxin production and immunity to the same quorum switch so non-contributors get punished. Hosts and competitors fight back too. Seaweeds like Delicia pulchra release molecules that jam bacterial signals. Many microbes make enzymes, such as lactanases and achylases, that degrade AHLs. Researchers call this quorum quenching. Medicine is exploring the same idea. Instead of killing bacteria, dial down their ability to coordinate virulence and biofilm formation by interfering with signals, a strategy that could make infections more manageable and antibiotics more effective. There are limits and misconceptions worth clearing up. Quorum sensing is not a universal bacterial language. It's a family of related systems whose details differ by species and context. Thresholds depend on flow and geometry. Signals wash away in fast-moving liquids and build up in crevices and mucus. Responses may be graded rather than binary, and what looks like perfect synchrony often arises from many cells, each making a noisy decision that averages into a clean group response. In the lab, scientists harness these parts to make living circuits. Put the lux promoter upstream of a gene, and you've built a population-sensitive switch. Wire a signal in a loop, and you can synchronize oscillations across a colony so that cells flash in time. Route signals between strains, and you get task-sharing consortia that produce drugs or digest pollutants only when enough teammates arrive. All of this works because the logic is simple. Small molecules move, receptors read concentration. Gene networks translate that reading into action. If you want something concrete to picture, imagine a tide pool at dusk. A few scattered cells whisper and nothing happens. As the tide turns and the pool grows crowded, whispers stack into a chorus. The chemistry crosses a threshold, and 1,000 lanterns blink on at once. For you, the lesson is not a forced self-help metaphor, but a gentle observation about timing and context. Many systems, microbial, ecological, even social, delay costly actions until signals say the effort will matter, and that patience is part of intelligence. You don't need to memorize the names of signals to feel the underlying idea. Life invented a way to count without counting, to decide together without a leader, to move from quiet potential to shared action with a nudge as soft as a molecule drifting back home. When you finish listening tonight, notice how calm that is. No magic, no exaggeration, just tiny messages crossing a line, and a community quietly becoming more than the sum of its lonely cells. Protein interaction webs. Y shape means function. Hold a paperclip in your mind and twist it only slightly. How quickly it stops being useful, and you're ready to feel why. In living matter, shape is destiny. Proteins are the tiny tools your cells use for almost everything. And what each tool can do depends on how its long chain of building blocks folds into a three-dimensional form, whose bumps and grooves, soft hinges and rigid beams line up with partners like a key slipping into an irregular lock. That form is not sculpted by a micromanager but coaxed by the chemistry of the chain itself. The water around it pushes oily parts inward. Polar groups pair up. Side chains hunt for complementary pockets. And as the chain samples possibilities, it settles into a shape that works well enough to start binding, cutting, hauling, signaling, or guarding. Inside this world, fit is more than a metaphor. When two proteins meet, they don't clasp by magnetism or myth, but by a patchwork of small forces. Hydrogen bonds, van der Waals touches, salt bridges, distributed across an interface that often looks like a rugged coastline. A handful of hotspots on that coastline supply most of the grip, so mutations there can weaken a partnership dramatically, while changes far from the hotspots barely matter, which is why a single letter change in one gene can derail a pathway that usually depends on dozens of players. Yet shape is not a statue. It is a living compromise between stability and flexibility. Many proteins breathe, tiny motions open and close pockets, swing loops, or rock domains. So binding can follow two honest stories. Sometimes a partner nudges a protein into the right pose, induced fit. Sometimes the protein is already flickering through that pose and the partner simply chooses it, conformational selection. This breathing allows allostery, where binding at one site shifts the shape of a distant site to increase or decrease its readiness. The classic case is the oxygen-carrying protein in your blood, which takes on oxygen more easily after the first molecule binds and then releases it more easily where it's needed. But the principle is everywhere. Enzymes turn up or down, receptors prime their neighbors, channels widen or narrow, because information in biology is often a change in form passed along a chain of contacts. The rules grow richer when you notice that not all proteins choose a single resting pose. A large class called intrinsically disordered proteins remain flexible in isolation, acting more like soft strings than folded wrenches, and only adopt structure when they dock to targets. Their looseness is not sloppiness but strategy, letting them embrace many partners with short linear motifs that can be toggled by small tags. Phosphates, acetyl marks, and other modifications that add or subtract tiny bits of charge or bulk to switch a binding site on or off. These weak, multivalent contacts can also make proteins gather into liquid-like droplets inside cells, membraneless compartments where certain reactions run faster or are kept apart from others. Your nucleolus, stress granules, and other condensates behave this way, forming and dissolving as conditions change. And when the delicate balance of stickiness and repulsion is lost, droplets can harden into gels or fibers that cells struggle to clear, a path toward neurodegenerative disease that begins with ordinary shape and ends with stubborn tangles. Because shape matters so deeply, cells invest in guardians that help chains fold and refold correctly. Chaperones bind newborn or stressed proteins, shielding their sticky patches until the right contacts form. And barrel-shaped machines give misfolded guests a private room to try again. When folding fails, quality control tags direct the rejects to the cell's grinders for recycling, an economy of parts that keeps function sharp. When folding goes wrong and escape routes are blocked, misfolded proteins can seed others to misfold, self-propagating errors at the edge of chemistry and catastrophe, reminding you that precision is both a power and a risk. If you zoom out from one clasp to the whole room, proteins form interaction webs, interactomes, where a few hubs touch many partners and countless other nodes play quieter, specialized roles. Some hubs are scaffold proteins that don't catalyze reactions themselves, but hold the right pieces close together so signals move efficiently and crosstalk stays organized. Like architectural brackets in a busy control room, in neuron synapses, scaffolds line up receptors, enzymes, and structural parts so that incoming signals are translated into changes in sensitivity. In growth pathways, interchangeable domain types, SH2, SH3, PDZ, and friends, recognize small sequence motifs, letting evolution shuffle modules into new circuits without reinventing physics. The language here is affinity and specificity. A bond must be strong enough to hold for the job, yet weak enough to let go on time, specific enough to prefer the right partner, yet tolerant enough to function amid noise. Cells tune this balance with local concentration, cooperative binding, and those tiny post-translational tags, so a protein can behave differently at the membrane than in the nucleus, or at noon than at midnight. Medicine and engineering live at these interfaces. Traditional drugs often fit into deep pockets on enzymes, but many crucial disease proteins lack such cavities, or they harm by forming the wrong partnerships rather than by catalysis that has pushed discovery towards surfaces once thought undruggable. Targeting hotspots at protein-protein interfaces, luring partners apart with cleverly shaped molecules, or recruiting cellular disposal machinery to tag a harmful protein for removal, rather than trying to block it head-on. The rise of high-resolution tools has made this practical. Cryo-electron microscopy images massive complexes in near-native states. Crystallography and NMR illuminate smaller dynamic interactions, and chemical cross-linking and single-molecule methods capture fleeting touches that used to escape detection. Machine learning that predicts static structures from sequence has accelerated thinking, especially for parts that do fold cleanly. But it is worth remembering that a static structure is a starting frame, not the whole film. Interfaces breathe, modifications come and go, and crowded cellular environments shift preferences that look simple in a purified tube. If you want a feel for evolution's hand, notice how domains get reused. A grip that once bound a signaling tail in a primitive organism is redeployed in your cells to wire entirely new circuits, and small mutations that change the depth of a pocket or the charge at a rim retune an affinity just enough to switch partners or timing. Constraint and creativity dance here, with essential hotspots guarded by purifying selection, while peripheral ridges and loops drift, inviting novelty without destroying the core. Even across species, the same fold, an ancient barrel or sandwich, reappears, carrying different decorations that specify new tasks, a reminder that life builds complexity by swapping and tinkering with shapes that already work. The honest takeaway, as you let the day slow, is wonderfully concrete. There is no mysticism in why a protein matters. Its usefulness resides in a form honed to fit other forms, its reliability in networks of contacts strong enough to carry action and soft enough to reset, its pathologies in small deformations that ripple across partnerships and your health. Memory and movement depend on this choreography every second. When you face complexity in your own choices, you might borrow the protein's wisdom. Keep a sturdy core but allow flexible loops. Use a few well-placed hotspots to anchor what matters. Let weak ties assemble and disassemble as conditions change. And remember that the right shape for today's task may not be a rigid ideal, but a family of poses that can breathe with the moment. In that breathing lies function, and in function, the quiet meaning of form. Microtubule highways inside the cell's logistics. If you could shrink down and walk the length of a neuron, you would find not chaos, but a disciplined freight system humming under soft light. Long, hollow rails stretching from the cell's core to its distant edges, motors stepping with delicate certainty, and packages labeled for exact delivery. And the more closely you look, the more obvious it becomes that microtubules are the cell's logistics network, built for speed, reach, and timing in a space where getting cargo to the right place at the right moment makes the difference between thought and silence, growth and drift, health and disease. A microtubule begins as a line of identical parts, tubulin dimers, stacked head to tail into stiff protofilaments and wrapped into a tube. But that tube is not symmetrical. It has a plus end that usually grows faster and a minus end that grows more slowly, or anchors at a nucleation site, often a centrosome in animal cells studded with G-tubulin rings that seed new rails. This polarity turns every microtubule into an arrow, and the cell reads those arrows to decide which way traffic should flow. The tracks are never fully settled because a built-in instability keeps them honest. New dimers carry a molecular cap that favors growth, but if addition slows and the cap erodes, the end can peel back in a sudden loss called catastrophe. Growth resumes when a fresh cap forms. And this cycle of growth and shrinkage, dynamic instability, lets a cell explore space quickly, sampling directions until a connection matters. Watch this with fluorescent tags and you'll see bright comets at growing ends as proteins like EB1 ride the newest lattice. In a dividing cell, hundreds of such ends radiate, searching for kinetochores on chromosomes, and once they latch, tension and error correction enzymes work together until each sister chromatid is attached to the proper pole, a quiet miracle that repeats each time a body is made. The network is valuable because tracks alone do nothing. The power lies in motors that read polarity and walk. Kinesins, a large family of two-footed proteins, usually head toward plus ends, hauling secretory vesicles, membrane components, and even mRNA packets outward to the cell's periphery. Dynin, a heavier ring-shaped motor, tends toward minus ends, carrying worn-out materials and signals, back toward the center and positioning organelles near the nucleus with the help of a scaffold called dynactin and adapters like LAS1 or BICD2. Each kinesin step is a hand-over-hand cycle powered by ATP, one head binding a head while the other releases, and each dynin stride involves a coordinated shift in its ALA plus ring and a swinging stalk that grips and lets go. The result is processive movement over micrometers, which at the cellular scale is the distance between a shipment missed and a life saved. Nowhere is this more vivid than in nerve cells. An axon can run a meter in your leg, and without robust anterograde transport, synaptic terminals would starve of enzymes and membrane. Without retrograde transport, signals from the periphery would never reach the body to trigger growth, repair, or, in the case of some viruses and toxins, a kind of hijacked surreptitious entry. Axons showcase another subtlety. Not all tracks are alike. Some microtubules are long-lived and acetylated within their hollow core. Others are newly made and more flexible. Some carry a chemical detour sign called detyrosination at their ends. Others are rich in polyglutamylation on the outside. And these small tags bias which motors prefer which rails. A tubulin code that gives the network a built-in routing layer. Dendrites add complexity by mixing track polarity, plus ends pointing both in and out, so traffic can be carefully segregated between incoming synaptic machinery and outgoing waste or signals, preventing crosstalk in a space where mistakes would fog thought. Even the Golgi and endoplasmic reticulum take their cues from this system. The Golgi normally huddles near the minus end-rich center. Its stacks shaped in part by microtubule tension, while the endoplasmic reticulum spreads along growing plus ends, hitchhiking to reach far-flung regions where calcium storage and protein folding are needed. In pigment cells, a simple demonstration of coordination plays out when hormones alter the tug-of-war between kinesins and dynin. Melanin granules disperse along plus-end tracks to darken a frog's skin or gather inward to pale it, changing an animal's shade in seconds by adjusting motor balance rather than making new pigment. During cell division, the network retools itself almost entirely. The centrosomes split and nucleate radial arrays that become the spindle. Microtubules probe and capture chromosomes. And force arises in more than one way. Motors sliding antiparallel microtubules apart push poles away from each other. Enzymes that specifically peel dimers off ends create a kind of Pacman depolymerization that reels chromosomes inward. And cross-linkers act as shock absorbers to keep the system from tearing itself apart under load. Receptors on kinetocores sense tension and summon repair. When two microtubules attach from the same pole to both sisters, an error, and a checkpoint halts division until the geometry is right. It sounds formal, but it's material science. Grips, springs, pulleys, and safety catches played out in protein. Microtubules also build hair-like organs that sense and move. Cilia and flagella are specialized bundles with a 9+2 microtubule architecture lined with dynin arms that try to slide neighboring doublets past each other. Because spokes and links constrain them, sliding turns into bending, and coordinated waves propel cells or sweep fluid across tissues, clearing airways, moving eggs along the oviduct, and letting certain cells taste their environment with a single, non-motile cilium loaded with receptors. Keeping these organelles healthy requires intraflagellar transport. Kinesin 2 moving cargo to the tip, dynin 2 returning it, and when this assembly line falters, a spectrum of ciliopathies emerges, from respiratory trouble to kidney cysts to subtle defects in left-right patterning during development. For all this activity, stability must be tuned, and the cell does this with a toolkit of binders and remodelers. Some proteins, like XMAP2 and 15, act as polymerases that speed addition at tips. Others, like kinesin 13 family members, are specialized depolymerases that curl protofilaments and induce shrinkage. End-binding proteins track new growth and recruit partners. Structural maps, such as TAU in axons or MAP2 in dendrite space, and stabilize tracks differently, so compartments maintain identity. When control slips, tau detaching or aggregating, modifications skewing balance, axonal transport degrades, traffic jams build, and neurons falter, a link that helps explain why transport defects show up early in degenerative conditions. Medicine has long exploited the vulnerability of this network in fast-dividing cells. Drugs like paclitaxel freeze microtubules in a stubbornly stable state so the spindle cannot rearrange, while vinca alkaloids destabilize assembly so tracks never form properly. Both strategies are arresting mitosis and pushing cancer cells toward death. In gout, colchicine calms destructive inflammation by interfering with microtubule-dependent cell motility. And in the lab, reversible inhibitors like nocodazole let researchers switch tracks off and on to test causality in living systems. Plants remind you the design is universal but adaptable. They lack centrosomes, yet lay down beautiful cortical arrays of microtubules just under the membrane that guide the angles of cellulose fibers, controlling how cells elongate. Before division, they briefly assemble a preprophase band that marks where the new cell wall will fuse. And after chromosomes separate, a phragmoplast of microtubules directs vesicles to build that wall precisely, a logistics variant tuned for life in a stiff box. Through it all, microtubules also bear mechanical load, resisting compression as actin filaments pull in tension, giving the cell a resilient scaffold. Motors pushing on tracks generate not just motion but force, which cells sense and answer by altering growth direction or stability, a local feedback that aligns logistics with geometry. It is tempting to imagine such order requires a planner, but the truth is less romantic and more satisfying. Polarity emerges from how subunits add, exploration from instability, direction from motors that evolve to favor one end, routing from small chemical tags and local helpers, robustness from numbers and redundancy. And the conclusion, as the room goes quiet, is gentle. Inside you, there runs a patient freight system that rarely boasts, a lattice that grows, retracts, and grows again, cranes that lift and set down without clanging, packages that carry permission, repair, memory, fuel, and schedules that adapt by feel rather than decree. Proof that life's clarity does not depend on perfect stillness or rigid plans, but on rails that can be rebuilt in motion and couriers that know how to find their way home. Cytoskeletal tensegrity, architecture as strength. You've been told that cells are sacks of chemistry, but picture them instead as tiny suspension bridges that never stop adjusting their cables. Because one of the most powerful ideas in modern cell biology is that your cells are built on tensegrity, a balance of continuous tension and isolated compression that turns soft matter into architecture. And once you feel that balance, countless mysteries about form, strength, and even gene control begin to make sense. In a tensegrity structure, cables carry pull and struts carry push. In you, actin networks and myosin motors generate contractile tension throughout the cytoplasm. Intermediate filaments distribute and buffer that tension so it doesn't snap. And stiffer elements, bundled actin, thickened microtubules, densely packed extracellular matrix fibers, act like compression-resisting struts that keep the whole volume from collapsing. The cell stays poised in a pre-stressed state, always slightly taut, so it can react in milliseconds to a nudge without tearing or having to rebuild from scratch. That pre-stress flows outward through focal adhesions, molecular, anchor plates where integrins span the membrane and link inside to actin and outside to collagen or fibronectin. So when the substrate is stiff and well organized, the cell can pull against it and strengthen its own scaffolding. When the substrate is soft or disordered, the same tug yields little resistance and the cytoskeleton relaxes, changing shape, stiffness, and downstream signaling. This simple mechanical conversation has deep consequences. Stem cells, for instance, tend to adopt neuron-like fates on gentle, brain-soft gels and bone-like fates on stiffer matrices, not because the gel contains secret instructions, but because tension and adhesion recruit different biochemical pathways that sway the same genome toward different outcomes. Within minutes of a change in tension, mechanosensitive proteins at adhesions unfold to reveal docking sites. Kinases like FACA light up, ROA rock tone the cables, and within the nucleus, the YAP TAS coactivators shuttle in or out, depending on how taut the system is, nudging whole gene programs toward growth, migration, or quiet maintenance. Force doesn't stop at the membrane either. A molecular truss, the link complex, ties the cytoskeleton to the nuclear lamina so that a tug on the surface can ripple across chromatin, altering how accessible certain stretches of DNA are, which means mechanotransduction isn't a buzzword, but a physical route from touch to transcription. When lamins are mutated, as in progeria, the nucleus stiffens and tears more easily. Cell mechanics go awry and gene expression drifts, a vivid reminder that the integrity of your architecture helps set the tempo of your biology. Tensegrity is also a language of shape under stress that you can watch with your eyes. Endothelial cells lining blood vessels elongate and align with flow, redistributing tension in ways that lower inflammatory signaling where flow is smooth and increase it where flow is disturbed, which partly explains why plaque tends to collect at branched, chaotic regions. Red blood cells, which lack a nucleus altogether, maintain their springy disc through a spectrin-actin mesh under the membrane that works as a tensile net against the compressive squeeze of capillaries, and when that net is altered, cells stiffen and break or clog. Fibroblasts in connective tissue pull the matrix into tracks and ridges as they migrate, each adhesion bearing load like a footfall on a rope bridge. And the amount of tension they maintain can determine whether wounds heal into flexible skin or into dense fibrotic scar. Because tensegrity is a principle rather than a part, it scales. Your tendons and fascia are pre-tensioned cables, stabilized by compressive bones. Your lungs' alveoli stay patent through a balance of surface tension and connective tissue springs that distribute pressure. Even at the whole organ level, the heart's muscle fibers and the collagen scaffold share loads so that contractions don't cave chambers in the wrong places. Disease often arrives as a mechanical misreading. Cancer cells commonly remodel their cytoskeleton and adhesions, lowering their internal pre-stress so they can squeeze through basement membranes and soft tissues. At the same time, the tumor microenvironment stiffens as collagen crosslinks accumulate, which perversely feeds growth signals back through integrins and keeps the malignant loop running. In chronic fibrosis, myofibroblasts crank up actomyosin tone and lock the matrix into a rigid, scarred state, making it even easier for cells to maintain high-tension signaling. One reason why breaking that feedback with drugs that relax the cytoskeleton or soften the matrix can ease disease. In the nervous system, too much tau detaching from microtubules or too little cross-linking support can destabilize structural balance and stress transport, setting the stage for failure long before neurons die. Even immunity has a mechanical accent. If epithelial cells lose proper adhesion and tension, they can trigger alarm pathways or undergo anoikes, a form of cell death on detachment that prevents misplaced growth. Architecture here is surveillance. Tensegrity explains everyday lab observations that used to look like trivia. Put the same cells on a thin, bendy glass cover slip versus a thick, rigid one and watch their spread area. Stress fibers and nuclear YAP change. Pattern adhesive islands into small squares and cells round up into long rectangles, and they build muscular cables along the long axis. Change the alignment of fibers in the matrix, and migration follows those lanes by durotaxis, a preference for paths that keep tension coherent. The beauty is that no central controller is needed. Proteins that sense force unfold when pulled. Enzymes that feel packing density change their pace. Ion channels such as piezo open under membrane tension to adjust calcium influx. Each local response is small, but because the cytoskeleton is continuous, the entire cell hears the news and settles into a new balance point. This same continuity is why small manipulations can have surprisingly global effects. A tug on a single adhesion bead with a microneedle can stiffen the whole cell within seconds, and relaxing myosin just a notch can soften the nucleus by measurable amounts because the cables slacken and compressive struts no longer bear the same load. Clinically, we already exploit the idea, sometimes without naming it. Orthopedics relies on Wolff's law, the bone's habit of reinforcing along lines of stress, to strengthen skeletal architecture through graded loading. Physical therapy and deep tissue work remodel fascia and muscle tone to redistribute tension, so joints find neutral. Even the success of certain antifibrotic drugs, Rho kinase inhibitors, and contact-activated stents can be read as tuning a tensegrity network back into a safer range. In the lab, microfabricated pillars that bend under cell traction let us measure internal pre-stress. Optogenetics that locally switch on contractility let us watch a wave of stiffening propagate. Engineered matrices with tunable stiffness, viscosity, and fiber alignment disentangle which mechanical cues cells trust most. The honest limits are instructive too. Tensegrity isn't the only story, and not every tissue behaves like an idealized web, but as a scaffold for thinking. The model earns its keep because it predicts how a small change in tension can cascade into a different identity, a different healing path, a different risk. Let the conclusion land softly. Your body holds together and decides its future, not just by the molecules it carries, but by how those molecules are arranged to share force. A quiet engineering wisdom that keeps cells ready, resilient, and responsive. If you borrow that lesson, you start to design your own life less like a brittle rod and more like a tuned network. Keep a little healthy pre-stress so you respond without breaking. Pair flexibility with a few well-chosen struts. And remember that strength is not the absence of movement, but the artful balance of pull and push that lets movement carry meaning. Viral host negotiations. Molecular arms races. Imagine your cells as quiet rooms where bargains are struck under pressure. An uninvited courier slips in carrying a minimal script. Your defenses demand credentials, and from that moment, a rolling negotiation begins in which each side rewrites the rules while the other is reading them. That is the essence of viral host arms races. Not thunderous battles, but precise contests over entry, detection, replication, and escape. And once you see the steps with clear eyes, the drama becomes both more grounded and more astonishing. A virus on its own is little more than instructions wrapped in protein, sometimes in a fatty envelope. To matter, it must touch the right lock on a cell, open the door, uncase its genome, and redirect the cell's machines to make more of itself. Your cells don't wait passively. They keep sentries that feel molecular patterns, sensors in the cytoplasm that notice odd RNA ends or misplaced DNA, and receptors in the endosome that recognize viral fragments. And when these trip, they launch interferon, a broadcast that flips thousands of antiviral switches in neighboring cells. Those switches aren't mystical. Some make it harder to build proteins from viral messages. Some shred viral RNA. Some block the assembly or release of new particles. And some mark infected cells for elimination. The negotiation starts here because successful viruses arrive with countermeasures already packed. Influenza's NS1 protein muffles the alarm by binding RNAs and meddling with the pathways that would otherwise amplify interferon, letting the first rounds of replication proceed in relative quiet. Ebola's VP35 and related proteins cloak viral RNA so the sensors fail to recognize it. Coronaviruses devote several genes to sanding down the cell's alertness. One early protein sits on the ribosome and subtly jams translation of host messages, while another impedes the traffic of protective signals into the nucleus. The result is not invisibility, just enough confusion to buy time. HIV makes a different bargain. It carries accessory proteins that directly sabotage specific restriction factors. Inside human cells, one viral factor tags a host enzyme for disposal because that enzyme would otherwise pepper the viral genome with disabling edits. Another pries away a tethering protein that would have pinned new virions to the membrane, and yet another drags down the immune ID badges on the cell surface so killer lymphocytes cannot easily spot the infection. This back and forth leaves signatures you can read in our DNA. The genes encoding such host restriction factors often bear marks of rapid change across primates, as if they were sprinting to keep up. In turn, viral proteins that bind them evolve fast, tweaking their shapes to stay compatible with yesterday's host variant and slippery to tomorrow's. You can see the same logic in receptor evolution. A well-known human variant that truncates a chemokine receptor on immune cells dramatically reduces the chance of HIV entry. In influenza, subtle differences in the sugars that crown airway cells can shift which species a strain prefers, shaping spillover risk. And across populations, the diversity of immune presentation molecules, those that display viral pieces to T cells, looks excessive until you see it as a hedge against any one virus learning a single cheat that works on everyone. Now step back and consider how viruses maintain the upper hand long enough to spread. Many RNA viruses copy their genomes with error-prone polymerases, generating swarms of related sequences inside each host. That cloud of variants, quasi-species, isn't just sloppiness, it's flexibility. Because under pressure from antibodies or drugs, the rare variant that survives becomes tomorrow's dominant form. You've met this in everyday news as drift, the incremental change that forces regular vaccine updates for fast-evolving viruses like flu, and as shift, the rarer reshuffling of whole genome segments when two strains mix in the same cell, yielding a version with a new combination of surface markers that few immune systems recognize. Hosts answer with diversity of their own. The machinery that builds antibodies deliberately mutates and selects binding sites during infection, honing sticks that fit the current foe more tightly. Memory cells linger for years to mount faster, stronger responses when a similar threat returns. Even bacteria play the game. Long before vertebrates evolved antibodies, microbes learned to keep a scrapbook of past viral encounters by storing short bits of phage DNA in clustered repeats, what we now call CRISPR, and to cut matching invaders on the next attack. Phages, unwilling to yield, evolved tiny proteins that jam those cutting complexes, and some went further by altering their own DNA chemistry so that bacterial restriction systems see the genome as self rather than enemy. None of this is a steady line of escalation. It is a tangle of feints and counterfeits. Some large DNA viruses make decoy versions of our own signaling molecules to soak up the alarm, or secrete fake receptors that catch interferon before it can warn neighbors. Others decorate their surfaces with dense forests of sugars borrowed from the host, forming a glycan shield that hides vulnerable protein features from antibodies. Even when an antibody binds once, a minor tweak in the shield can blunt the grip the next season. Some viruses avoid notice by going quiet. Herpes viruses slip into long-term latency in neurons, sheltering where immune patrols are less vigilant and reawakening under stress. Retroviruses embed a copy of themselves in host DNA, creating reservoirs that can outlast long treatment unless every last replicating niche is closed. And some manipulate the flow of time inside a cell, pushing the host to shut down its own mRNA translation, or to alter the pathways that transport immune messages, so that even if the alarm rings, help arrives late. From this realism grow our best medical strategies. Combination therapies against HIV and other chronic infections aim at multiple steps: entry, genome copying, processing, so that even a fast mutating virus has trouble finding a single path that evades all blocks at once. If one route closes, another is open, but closing 5 at once is harder. Antivirals that target the most conserved parts of polymerases can remain useful as variants change, and monoclonal antibody cocktails bind different sites, so escape would require multiple simultaneous adjustments. Some treatments sidestep the viral genome entirely by targeting host factors a virus needs, a receptor antagonist that denies entry, or a drug that nudges innate defenses to a more prepared state. But these require care because what helps thwart a virus can also disrupt ordinary physiology. Vaccines, especially those that can be updated quickly, tilt the negotiation toward us by priming memory cells against conserved features that are harder for a virus to alter without breaking itself. The more we learn from mapping which mutations allow escape with minimal cost, the better we can choose targets that corner evolution into dead ends. There is also space for unexpected truces. Pieces of ancient retroviruses are stitched into our genomes, and a few have been domesticated for good. One viral gene has been repurposed in mammals to help fuse placental cells, a startling example of yesterday's invader becoming today's essential tool. On the bacterial side, phages are returning as living drugs against antibiotic-resistant infections, not because they are gentle, but because their appetite for particular pathogens can be harnessed safely when chosen well. If you let all of this settle, the conclusion is both sobering and oddly calming. There is no final victory in molecular arms races, only periods of advantage shaped by how quickly each side can learn and by how well it can pay the costs of its tricks. The honest lesson for you is to respect speed and redundancy, to support the defenses that buy time, sanitation, vaccines, early treatment, and to remember that nature's negotiations are not won by bluster, but by small, well-chosen moves at the right interfaces. As you drift towards sleep, consider the quiet competence inside you. Cells that listen without panic, systems that balance attack with repair, and a memory of past encounters that allows mercy toward the self and vigilance toward what does not belong. In that balance, you don't need a myth to feel safe, just the steady knowledge that life has been practicing this conversation for billions of years and is very good at it. Predator-prey oscillations, the math behind ecosystems. Picture a meadow that seems peaceful from a distance. Grass is bending, small animals nosing through stems, a shadow drifting overhead. and then lean in far enough to notice the hidden rhythm that pushes the scene forward. When food for the plant eaters is abundant, their numbers rise, which tempts more hunters into the open. And as the hunters prosper, the plant eaters dwindle, which starves the hunters back. And with the hunters thinner on the ground, the plant eaters rebound. This seesaw is the heart of predator-prey oscillations, a natural cycle that repeats not with metronomic perfection, but with enough regularity that biologists and ecologists have been measuring it for more than a century. You can read the story in fur trade ledgers that tracked snowshoe hair and lynx pelts across years. The hair numbers swell. The lynx numbers rise a bit later. The hares crash. The lynx follow. And you can watch it in controlled arenas where tiny predators such as predatory mites chase equally tiny herbivores across orange peels or in glass flasks where a fast-growing microbe is hunted by a single-celled predator. If prey multiply quickly and predators respond with a delay, a wave forms. The simplest intuition is this: prey increase when they have food and space. Predators increase when they have prey. And neither keeps climbing forever because limits arrive. Plants run short. Winter bites, hiding spots fill, mothers tire. There is a characteristic lag that makes the pattern look like a dance. Prey crest first. Predators crest after. Prey dip. Predators dip later. And the timing shifts with biology. How fast prey reproduce. How quickly predators mature. How long it takes a full belly to become more mouths. But real ecosystems never read a single script. So the shape of the wave depends on how predators respond, which is biologists' shorthand for the relationship between prey availability and predator intake. Some hunters take food almost in proportion to how much is out there until they physically cannot catch more in a day. Think filter feeders or spiders saturating their web's capacity. Others slow down at high prey density because handling and digestion take time, creating a curve that rises quickly then flattens. Still others are inefficient at low prey numbers because prey hide well or switch habitats. So consumption ramps up only after prey are common. These response styles tilt the cycle. If predators overwhelm prey at low densities, crashes are deep. If prey enjoy refuges, burrows, thickets, or simply the maze of a complex habitat, the bottoms are not so low and the system steadies. Time delays matter just as much. A fox that eats well today does not produce cubs tomorrow. A fish that sees a bounty of small fry needs months to turn that into a larger adult population. Delays can turn a gentle approach to balance into oscillations, and, when the delays are long and gains very strong, even chaotic swings, where the next peak is impossible to guess from the last. Seasonality layers in a drumbeat. In northern forests, plant growth surges in spring. Herbivores pulse in early summer, and predators follow. A late frost or a massed year, when trees flood the forest with seeds, can amplify or mute the cycle by changing the bottom-up supply of energy. Top-down forces pull the other way. Add an extra level of hunters above the usual predators, and the pressure on herbivores increases. Remove those apex hunters, and the middle predators explode, often hammering the small herbivores and altering the vegetation in a ripple called a trophic cascade. You can see these principles play out island by island and lake by lake. On Isle Royale, wolves and moose have traced long, imperfect loops across decades, with harsh winters, disease, and occasional migrations tipping the sizes of each population. In African savannas, migratory patterns of herbivores and the presence of large carnivores braid cycles with the rains. In the lab, early experiments often failed to produce stable cycles because the arena was too simple. Predators ate the prey to extinction or starved when prey hid too well. Only when researchers added refuges, uneven terrain, or periodic immigration did long-lived waves appear, a strong hint that real landscapes dampen extremes by providing patchiness and rescue effects. There is a caution here that teaches humility. The math behind ecosystems is not there to reduce nature to a toy. It helps you ask better questions. What slows predators when prey are scarce? What speeds prey when hunters rest? What background pressures, like disease or competition with other species, siphon energy from the loop? Consider the paradox of enrichment, a famous theoretical result later supported by experiments. Improving the prey's food base can destabilize the system, leading to larger population booms followed by deep crashes. Because the predator population swells in response and overshoots, a warning that feeding a system indiscriminately can make it fragile rather than safe. Fisheries management has lived this lesson painfully. Harvest a predator too hard and the prey can explode, overgraze their own food, and then collapse, taking the fishery with them. Harvest the prey too hard, and predators starve until only a few survive in poor condition. In both cases, the missing rhythm is replaced by a ruinous lurch. Early warning signs that a system is sliding toward a bad turn are becoming practical. As resilience wanes, populations often show critical slowing down, taking longer to recover from small disturbances. Variance and spatial patchiness can increase. Time series data thicken with certain kinds of flicker. All hints that managers can use to step back from the edge, reduce pressure, restore habitat complexity, or reintroduce missing predators to reknit the web. One reason these cycles feel intuitive is that they embody trade-offs that experience teaches you even outside ecology. Specialization versus flexibility. A predator that switches prey when one is rare is more stable. Short-term gain versus long-term stability. Maximizing a single season's growth can set up next year's bust. And the value of buffers, refugees, diversity of prey, heterogeneity of microhabitats that keep shocks from propagating. Parasites and pathogens add another layer. A prey population that booms may become a petri dish for a disease that then suppresses it independent of predators, which in turn starves hunters and lengthens the low phase. A predator stressed by toxins or heat may hunt less efficiently, allowing prey to overshoot and damage vegetation in ways that echo through soil and stream. Climate change threads through all of this by shifting phenology, the timing of births, migrations, bloom, and when predator and prey fall out of temporal sync, a cycle can wobble, even if numbers seem healthy. The gift of this framework is not that it predicts exact counts, nature is too textured for that, but that it highlights levers that matter, response curves, delays, refuges, background mortality, and the links between energy at the bottom and pressure at the top. If you want to test the ideas where you live, look for proxies. Vole populations swelling after seed-heavy years, and raptors cresting after, insect outbreaks following warm springs, and the birds that chase them peaking later. Or in your garden, ladybirds arriving just a bit after aphids surge, then floating away once the feast is over. When you hold the picture gently, the conclusion is both practical and calming. Ecosystems breathe through relationships, not through dominance. Stability is a rhythm, not a straight line, and wise care often means leaving enough complexity, enough shelter, enough diversity, enough slack, that the next disturbance is absorbed rather than amplified. Carry that with you as the day quiets. You don't need equations on the page to feel the math in motion, only the patience to watch crests and troughs roll through the living world, and the humility to guide with a light hand so the wave can keep returning, not because you forced it, but because you learned how it moves. Food webs, energy, that climbs and loops. Start with a taste you barely notice. The cool sweetness of air in your lungs came from a leaf turning light into sugar, and that sugar is the first step of a climb. Energy captured, packed into carbon bonds, and handed from one eater to the next until it fades as heat. So when you look at a meadow, a tide pool, or a city park at dusk, you're really watching a braided economy where energy ascends through feeders and then loops back through the dark work of decay, a web more intricate than any ladder you learned in school. Plants, algae, and photosynthetic microbes are the entry gates, fixing sunlight into matter that can be eaten. In some corners of the planet, bacteria at hydrothermal vents do the same job using chemical energy from the earth instead of light, proving that webs do not require sky to begin, only a way to charge the world's batteries. Herbivores take their share, predators take theirs, and at each handoff, most of the power is spent on life's housekeeping, moving, growing, staying warm, staying ready, so only a modest portion passes upward. That is why a forest can support countless insects and a choir of small birds, but only a few large hawks, and why big hunters range so widely to feed. The simple pyramid you picture, plants at the broad base, then grazers, then carnivores, is real enough to orient you. But the lived web is messier and more resilient because the lines cross and loop. Omnivores graze and hunt. Many fish shift diet as they grow, and even plants are not purely givers, because some trap insects or live in partnerships that trade sugars for protective services. Beneath the visible traffic hums the detrital route, where fallen leaves, dead plankton, shed skins, and lost feathers become food for decomposers. bacteria and fungi unspool the stored energy and unlock nutrients, while earthworms, springtails, and a whole cast of scavengers chop debris into pieces small enough for microbes to finish the job. In the sea, an especially elegant detour, the microbial loop, reclaims dissolved organic matter that would otherwise drift away. Free-living bacteria capture it, tiny protists eat the bacteria, zooplankton eat the protists, and the energy rejoins the larger web. This quiet rescue explains how seemingly clear, nutrient-poor waters can still fuel a bloom of life. You can feel how structure shapes function when you look at real places. In a kelp forest, towering algae gather light, sea urchins graze the holdfasts, and sea otters keep the grazers in check. When otters vanish, urchins can mow the forest into a barren plain, and the web collapses to a simplified, poorer state. In western streams, salmon feed in the ocean and carry marine nutrients back inland as they return to spawn. Their bodies feed bears, birds, and microbes. And even the trees along the banks borrow that marine nitrogen to build new leaves. A reminder that food webs are not fenced in by habitat edges. On tropical savannas, seasonal rains pulse energy into grasses, herbivores swell in number, and lions and hyenas crest after. When the dry season returns, carcasses move the flow downward into dung beetles, vultures, and bacteria, making room for the next surge. Those examples tempt hierarchy, but the deeper lesson is network. Each species is a node, each feeding relationship a link, and the pattern of links, some strong, many weak, determines how shocks are absorbed. A web full of weaker ties tends to be steadier because no single interaction whipsaws the system. Occasional strong ties can be crucial, anchoring functions like top-down control. But too many strong and narrow dependencies make a web brittle, a structure that looks robust until a few keystones are pried loose. Keystones are not always the animals with sharp teeth. Sometimes they are unassuming grazers that keep algae from smothering corals, or fungi that set the pace of decomposition, or a parasite that quietly links hosts and shifts who thrives. Add parasites and the number of links in a web often doubles, because every host becomes both habitat and food, an uncomfortable but honest expansion that changes how energy moves. Chemistry matters as much as calories. Consumers need not just carbon for fuel, but the right balance of nitrogen and phosphorus to build proteins and membranes. Algae grown thinly on nutrients can be like salad with no dressing, filling but not nourishing, and grazers adjust by eating more, changing diet, or, when they cannot, by growing slowly and reproducing less. External subsidies complicate the picture. Wind-blown insects feed fish in mountain lakes, leaf litter feeds stream invertebrates that feed trout and kingfishers, and coastal upwelling brings deep nutrients to the sunlit surface, igniting blooms that echo through anchovies, seabirds, and whales. These side doors can stabilize a web by smoothing lean times, or destabilize it by inviting booms that crash when the subsidy fades. Humans redraw webs at speed. Heavy fishing can pull top predators down first, encouraging fishing down the web as fleets shift towards smaller, faster breeding species. Farms and cities leak nutrients that fuel algal blooms, which, when decomposed, strip oxygen and create dead zones where animals cannot breathe. Contaminants climb the web, concentrating in fat as they rise, so a toxin barely detectable in water can reach dangerous levels in a raptor or a seal. Invasive species splice in novel links, like a snake on an island that erases birds, cutting seed dispersal and changing the very vegetation that once anchored the web. Yet webs can heal when links are restored. Remove a dam and salmon may return, bringing back their nutrient ferry. Reintroduce a missing predator, and the middle layer may shrink to a healthier size, letting plants rebound and riverbanks knit themselves. Rebuild wetlands, and a filigree of detrital roots soaks up floods of nutrients and slows their rush to the sea. If you want to think like a caretaker rather than a conqueror, you ask web-shaped questions, not how many deer, but who eats whom, when, and with what side effects. Not how big is the harvest, but how many links does this cut, and do backups exist? You look for early warnings, simplifying diets, shrinking age classes of predators, seasonal pulses that arrive out of sync with the young that need them, because those signals whisper about frayed ties before collapse. You also notice how your ordinary acts lean on the web in both directions. Compost that returns nutrients to soil, shade trees that temper stream temperatures so oxygen stays high, coastal vegetation that slows runoff so blooms don't tip into disaster. The science here is clear and kind. Energy climbs through living ladders that rarely stand alone. It loops through decay and returns as fuel. And it sweeps sideways across boundaries that only maps insist are firm. The stability you want is less a fixed picture than a moving braid that can flex without snapping. Let that land softly as the evening quiets. Every meal you take is a handshake with a web of strangers, leaf and grazer, hunter and scavenger, fungus and bacteria, and the gratitude that rises when you see those relationships clearly is not sentimentality, it's literacy. You are part of a conversation about how energy becomes life and then returns. And your best choices are the ones that keep the climb steady and the loops unbroken, so the voices you love, bird song, surf, the breath of a sleeping child, have power to continue. Social contagion, ideas that spread like genes. You felt it without naming it. One person in your circle tries something new. You shrug, another mentions it a week later, and then a third, someone you trust for reasons that rarely make it into words, brings it up again, and suddenly the idea feels less like a stranger and more like a guest you've already met. This is social contagion, the way behaviors, beliefs, and moods move across human networks, sometimes like a cold needing only a single cough, sometimes like a custom that only sticks after repeated nods from different directions. And the science behind it is specific enough to be useful once you slow down to see the pieces. At its core, diffusion depends on two things you can picture: the kind of thing being shared and the structure of the network it has to cross. If a behavior is easy, low risk, and individually rewarding, clicking a link, trying a harmless snack, a single exposure can be enough, so the pattern resembles a spark jumping along dry grass. But if the behavior is costly or socially sensitive, changing your diet, joining a protest, adopting a new work tool that will make you stand out, you usually need multiple, independent exposures before you act. Reinforcement that tells you, people like me do this and they don't pay for it. Which is why researchers talk about simple versus complex contagion. The map matters just as much. Some people sit at hubs tied to many others. They can accelerate spread for easy things because a single mention touches many ears. Clustering, the tendency of your friends to also be friends, slows simple contagions by wasting repeated exposures on the same neighborhood. But it can supercharge complex ones by providing exactly the social reinforcement they require. Bridges, weak ties that link separate clusters, carry novelty into new neighborhoods. But unless those neighborhoods have their own internal support, complex behaviors often die on arrival. A lone adopter across a bridge is brave but lonely. This is why the same platform can launch a throwaway meme in hours and still struggle to move a careful habit across communities for months. The first is a spark in dry grass. The second is a fire that needs a hearth. You also need to separate contagion from its cousin homophily, the human tendency to cluster with people like ourselves. If friends in a gym class all start running more, it might be because they influence one another, or because running drew them together in the first place, or because the weather improved and everyone's mood lifted at once. Good studies try to tease these apart by looking at timing, who changed first, by using random nudges, assigning an early access feature to some but not others and watching if it spills over, or by analyzing network geometry. Influence tends to travel along certain paths while homophily is more symmetric. The practical point for you: don't call everything contagious just because it is shared. Look for directional clues and small experiments that make causation visible, then design accordingly. What actually travels? Not only facts and behaviors, but also frames, interpretations about what actions mean. Social proof, people like you did X, is especially potent when uncertainty and risk are high. Identity cues can lift or sink adoption before any argument lands. Emotion adds thrust. Messages that provoke awe, anger, or anxiety are more likely to be shared, and groups can synchronize mood in minutes through tone and posture. So an anxious crowd makes anxiety more likely even without new information. Memory and forgetting shape the wave too. If reminders are spaced so that each arrives just as the prior one fades, adoption rises with surprisingly little force. If exposure floods early and then vanishes, behaviors spike and collapse, a pattern you've seen in fleeting challenges that leave no trace. Platform design and policy tilt the board further. Recommendation systems that prioritize engagement will amplify items that trigger strong reactions, sometimes privileging controversy over accuracy. Quote, tweet, and duetting features lower the cost of rebroadcast. Friction, an extra click, a warning, a delay, acts like a speed bump, reducing impulsive spread without silencing anyone. Small interface choices become public health tools when what's spreading is harmful, and they become public education tools when what's spreading is helpful but effortful, like disaster preparedness steps. Contagion can be nudged ethically. Pre-bunking teaches common manipulation tactics before people encounter them, making later falsehoods feel familiar in the right way. Recognized, not believed. Prompts that ask users to think briefly about accuracy before sharing reduce the spread of low-quality headlines without heavy-handed moderation. Seeding through bridge figures, trusted by multiple clusters rather than merely popular, gives complex behaviors the reinforcement they require on both sides of a boundary. If you want to change a workplace habit, you can pick a handful of respected peers in each team rather than a single influencer. Make their actions visible to each other, and provide gentle public milestones so reinforcement accumulates. For community health, you can pair clear, low-effort actions with stories from diverse messengers so that recognition beats resistance. It's also worth naming ethical limits. People are not pipes and consent matters. Design that supports reflection and opt-out respects autonomy better than tricks that chase raw spread. The vulnerabilities of Contagion mirror its strengths. Echo chambers form when clustering becomes too tight and bridges are cut. Within them, complex beliefs feel safe because they are constantly reinforced, and contradictory facts struggle to enter because they arrive as isolated exceptions rather than as a chorus. Cascades, rapid, self-reinforcing waves, can carry truth or falsehood. The difference often lies in early conditions and available correctives. A community with many cross-cutting ties and norms for politely testing claims will see fewer wild cascades than one with brittle identity fault lines, because corrections spread as easily as errors. Diversity of connections is not a slogan here, it is infrastructure. It ensures that no single narrative can capture every channel at once, and it gives new, good ideas room to find multiple starting points. You may also want to remember that going viral is not the only goal worth having. Some changes are better built like coral reefs, layer by layer with room for repair, than like fireworks. For complex normative shifts, depth beats speed. You are not only trying to introduce a behavior, you are trying to build the local support that will keep it from being punished. That means designing for rehearsal and reward inside clusters, not just for reach. It means honoring early adopters who take reputational risk, not treating them as disposable launch pads. If you are the recipient rather than the instigator, learning to feel exposure counts is a form of literacy. When the third independent source brings you the same suggestion, you are not necessarily seeing a conspiracy. You may be seeing a threshold being crossed. When the only voices endorsing a claim are tightly interlinked, you may be hearing an echo, not a chorus. Simple habits help. Pause before sharing, add context when you do, check whether someone outside your cluster holds the same view, and keep a few bridges alive to people who do not think like you so that your attention is watered by more than one stream. As the day slows, let a gentler thought settle. Humans are not merely vectors, we are editors. We choose what to pass on and how. We can cool a feverish rumor by adding light rather than heat. We can warm a good practice by lending it our calm. And we can design our small corners, group chats, meetings, classrooms, so that repetition signals reliability only when it arrives from varied, honest sources. Ideas may spread like genes, but unlike genes, they ask for your stewardship at each step. And when you care for that responsibility, the networks you live in become less like dry grass waiting for a spark and more like tended gardens where growth continues, because you chose what deserved to climb and what needed, kindly, to stop. Urban metabolisms. Cities as living organisms. Walk a city at first light, and you can feel it before you can name it. The low thrum from buried pipes, the hum building in the power lines, the faint warmth lifting from yesterday's stone. And if you hold that attention a little longer, you start to see a living pattern. Cities eat, breathe, circulate, store, and shed. And the study of those flows has a plain name with profound consequences. Urban metabolism, the accounting of energy, water, materials, and wastes as if the city were an organism whose health depends on what it takes in, how it transforms, and what it lets go. In simple terms, a city draws high-quality energy, electricity and fuels, food and goods, embodied with their own hidden energy and water, and fresh water across long supply lines. Inside, those inputs are converted into heat, motion, light, meals, buildings, and services. Then the city exhales carbon dioxide, waste heat, wastewater, solid waste, and diffuse pollutants. Some of what comes in is not immediately used. It becomes in-use stock: steel beams, copper wiring, concrete, plastic, glass, the standing mass that stores past decisions the way a body stores calcium in bone. The numbers are sobering and clarifying. Although cities cover a sliver of land on Earth, they account for a large share of energy demand and emissions, because density concentrates both needs and opportunities. A single neighborhood can import power generated hundreds of kilometers away, water pumped over mountains, food grown on plains or hauled from oceans, and consumer goods whose supply chains circle the globe, which means what you see on your block is the last meter of an immense metabolic reach. Metabolism has quality as well as quantity. The grade of energy matters, because electricity can do delicate work while low-temperature heat is good for warming space or water. Matching the right job to the right grade is like feeding protein to muscle and fiber to the gut. Both are food, but they serve different needs. Waste heat from power plants and factories is a perfect example of misaligned diet. If you let it leak to the sky, it merely warms the air. But if you pipe it into district networks to heat homes or greenhouses, you drop the city's need for fresh fuel. The same principle guides combined heat and power plants, data center heat reuse, and low-temperature district loops that collect many small sources: supermarkets, ice rinks, sewage, and deliver comfort with far less waste. Water tells a similar story. It enters as treated supply, courses through homes and industries, and leaves with heat, nutrients, and contaminants. Those outflows are not gone. They are usable streams if you design for them. Modern plants can recover phosphorus as struvite crystals, a scarce nutrient that farms rely on, capture biogas from sludge to run generators, and polish water for reuse in cooling towers or irrigation. When you do that, the wastewater facility stops acting like a kidney that merely filters and becomes a liver that transforms. A shift from disposal to regeneration that cuts both imports and emissions. Storms reveal another layer of metabolism you might miss on dry days. In many older cities, rain shares pipes with sewage, and heavy downpours flush contaminants into rivers. By spreading green roofs, tree canopies, rain gardens, and permeable pavements, you give the city a way to drink, slowing, soaking, evaporating, so the pulse of a storm is absorbed instead of spiking the system. Trees are not just decoration here. Through transpiration, they move water back to the air, and through shade, they cut cooling demand, easing both the water and energy diets. Heat itself becomes a symptom you can read. Dark surfaces absorb sunlight by day and release it by night. Motors and compressors eject low-grade heat, and wind blocked by tall buildings lingers warm between walls. Together, these effects create urban heat islands that raise nighttime temperatures, stress vulnerable residents, and push air conditioners to work harder, a feedback that thickens the city's appetite exactly when you want to slim it. Cool roofs and pavements, more trees, and smarter layouts that invite breezes are small design choices that change a city's micrometabolism day after day for decades. Materials too deserve metabolic honesty. Every brick and beam carries embodied energy and emissions from its birth. Spreading a building before it has paid back that initial investment wastes more than money. Extending lifetimes, retrofitting for efficiency, and designing for disassembly turn the building stock into a flexible store rather than a landfill staged in slow motion. When structures finally age out, urban mining, recovering metals, glass, and even crushed concrete for new uses, closes loops that would otherwise stretch across continents. The most successful examples of industrial symbiosis show that this is not a dream. Cluster a power plant, a greenhouse complex, a cement kiln, and a chemical factory. And the waste steam, CO2, gypsum, and heat of one can be the feedstock of the next. Shrinking imports and exports simultaneously. Do the same at neighborhood scale by pairing a supermarket's steady refrigeration waste heat with an adjacent pool, or a server farm with housing, and you make metabolism local enough for ordinary people to benefit. All of this is measurable. Analysts use material flow analysis to tally tons in and out, input-output models to follow hidden footprints through supply chains, and stock flow maps to see how yesterday's construction becomes tomorrow's waste. When that bookkeeping is public, you have a dashboard instead of a hunch, and the political conversation shifts from slogans to levers: where to add storage, where to diversify sources, where a single failure would starve a district. The concept of scaling adds a hopeful wrinkle. As populations grow, many infrastructure needs increase slower than headcount because pipes and cables serve more people at once. If you plan well, each person can require less road length, less wiring, less line loss. But beware of rebound. Improving efficiency without changing habits or prices can make total demand climb anyway, because lower operating costs invite more use. Metabolic resilience means designing for buffers and diversity, not for knife-edge optimization. Multiple water sources, surface, groundwater, reuse. Microgrids that can island when the main grid stumbles. Storage that bridges daily and seasonal swings. And local production of some essentials, food, medicine, spare parts via fabrication, turn brittle lines into braided ropes. The boundaries of a city's metabolism are also ethical boundaries. A low-emission district heated by recovered industrial waste may look virtuous on paper while leaning on coal burned beyond its horizon. A fashionable neighborhood may boast farm-to-table menus but outsource manure, pesticide, and labor conditions to distant fields. A port may tally its local air quality gains, while the ships that feed it spew pollutants offshore. Honest metabolism widens the frame until the ledger matches the lived world, then narrows it again to ask, what can be changed close to home? That leads to practical steps you can feel. Align energy quality with task. Don't boil water with electricity if low temperature heat is abundant. Feed wastewater plants as resource hubs. Design stormwater as a harvest rather than a hazard. Price disposal so it reflects true cost and reward repair so it competes with replacement, and publish neighborhood-level flow maps so citizens can see where their efforts matter. If you are a planner, you can stage projects like a nutritionist stages meals, quick wins, LEDs, leak fixes, cool roofs that reduce cravings today. Medium moves, district energy, transit fleets, deep retrofits that change appetite. And long investments, compact mixed-use neighborhoods, shaded walkable streets, resilient waterfronts that reset the body's baseline. If you are a resident, small repeated choices, separating organics so biogas plants have feedstock, timing appliance use to align with cleaner power, supporting reuse and repair, are not drops in an ocean. They are local edits to the metabolism that spread when made visible and normal. And if you are tempted to dismiss the organism metaphor as cute, know that its value is practical. It pulls scattered departments, power, water, waste, street trees, housing, into a single systems view where a change in one organ shows up in another, where prevention is cheaper than cure, and where vitality is measured not by how fast the city can consume, but by how well it can circulate, recover, and thrive on less. Let the thought end softly. The city you love is not just a skyline, it is a pulse you can help steady. Cooler nights after hot days, cleaner water after storms, quieter rooms without sacrificing light. And when you listen for that rhythm and care for its flows, you're doing more than managing utilities. You're tending a living place so that it wakes rested, spends wisely, and breathes easier tomorrow. Trade networks, how flows create civilization. Open your palm and inventory. The quiet miracles resting there. A phone, a pen, a ring, a snack, and you are already holding a map of rivers you cannot see, ore lifted from one mountain and smelted on another, oil distilled into plastic halfway around the world, silica melted and cooled into glass, flavors carried across oceans by farmers you'll never meet. Trade networks are the hidden bloodstream that turns scattered resources into shared life. And once you learn to read flows instead of borders, you begin to see how civilizations are stitched together by movement. Goods, people, promises, and ideas riding the same paths at different speeds. The basics are deceptively simple, where local endowments differ. Tin here, timber there, fertile valley next door. Moving things creates gains for both sides. But movement costs time and risk. So the real story of civilization is the long, ingenious effort to lower those costs and tame those risks. Ancient caravans followed dependable water and friendly polities, building way stations and caravanserais that doubled as banks and news hubs. Monsoon winds became a seasonal engine across the Indian Ocean, teaching sailors to time departures like farmers time rains. Canals and roads weren't ornaments but arteries. The Grand Canal feeding China's capitals, Roman roads knitting provinces to a legal core. Because reliable throughput is the difference between a granary that feeds a city and a city that starves in a good year. Trust technologies then multiplied what a wagon or ship could do. Writing began as tally and seal. Standardized weights and measures turned haggling into contracts. Coinage compressed trust into metal, enabling strangers to settle accounts without kinship. Later, letters of credit and bills of exchange detached trade from the dangerous need to carry silver physically. So a bolt of cloth in one fair could become payment hundreds of miles away. Double-entry bookkeeping let merchants see profit and loss across time rather than by gut feel, and marine insurance spread the shock of storms across many shoulders, turning the ocean from a gambler's table into a calculable route. Every reduction in friction widened the map. Telegraph cables yoking grain markets on different continents so prices converged within hours rather than months. Refrigerated rail and ship holds moving meat and fruit across seasons and hemispheres. Containerization collapsing loading time from days to minutes by standardizing the box, quietly rearranging the geography of ports and factories because it became cheapest to bring the ship to the box and the box to any truck or train on Earth. If you look past the hardware, you see the network logic that makes some places boom and some wither. Hubs emerge where many routes meet, river deltas, mountain passes, deep harbors, and the bigger the hub, the more it attracts complementary crafts. Warehousing, finance, legal arbitration, repair. Bridges matter too, those slender links that connect otherwise separate regions, because they carry novelty as well as goods, which is why a small entrepot at a cultural edge can punch above its weight for centuries. But centrality is a double-edged advantage. Choke points, straits, canals, border rail junctions concentrate both power and fragility. A grounded ship, a border closure, a damaged bridge ripples through prices thousands of kilometers away, revealing how supply chains are really supply networks that gain flexibility from redundancy and break when those backups are shaved thin. You can feel the human layer in these flows. Merchants built norms that law later formalized. Fair courts in Champagne, guild rules along the Baltic, credit webs across the Mediterranean that Muslims called chirad and Italians commenda, long before nation-states could police every contract. Diasporas sustained trust across distance, translating languages and customs along the way. In many eras, women ran accounts and inventories while men traveled, an overlooked backbone to long-distance exchange. Each network also moved seeds, gods and germs, wheat and rice, potatoes and maize, coffee and tea, altered diets and populations. Faiths and philosophies rode merchants' caravans as often as armies' roads. Pathogens slipped into new hosts, remaking demography and policy, a reminder that openness requires hygiene and foresight, as well as courage. If flows build civilization, then information about flows, prices, inventories, delays, is its nervous system. When signals are timely and widely shared, arbitrage smooths shocks, and hoarding loses its edge. When signals are late or distorted, rumors become price, and panic does what drought alone could not. Modern platforms make that nervous system faster, but not automatically wiser. Real-time dashboards let a factory reroute purchase orders in hours, yet a miscalibrated algorithm or a stampede of just-in-time buyers can empty shelves as surely as a storm. That is why resilient trade looks less like razor-thin efficiency and more like good physiology. Buffers size to the hazard, diversity of suppliers and modes, transparent standards that make substitution safe, and local capacity for essentials, so communities can ride out temporary breaks without turning scarcity into conflict. Energy and materials deserve special honesty in this picture. Flows are not only pallets and parcels, but joules and atoms, copper and rare earths, phosphate and potash, oil and gas, increasingly electrons and data, the routes they travel, pipelines, high voltage lines, fiber optic cables, anchor alliances and anxieties, and their concentration in a few mines, straits, or factories can become leverage or liability. Moving from a fossil system to a cleaner one does not erase trade. It redraws it around metals, batteries, and grid gear, raising new questions about recycling, substitution, and the ethics of extraction. Meanwhile, the embodied flows in finished goods, water embedded in cotton, carbon in cement and steel, mean a city or country can seem light on paper while outsourcing heavy footprints. Honest accounting widens the frame to include those shadows, then lets policy nudge them toward cleaner sources, longer lifetimes, and circular loops where yesterday's scrap becomes tomorrow's feedstock. If all this sounds abstract, test it with one familiar object. Your phone's glass likely owes its clarity to sand from a few special deposits. Its chip carries patterns written in cleanrooms whose machines themselves are composites of global flows. Its battery mixes metals pulled from distant soils. Its software is a cultural product compiled across time zones. Its arrival depended on boxes sized to fit cranes built to fit ships, scheduled to pass canals dredged to fit hulls, widened by previous rounds of trade. And when you replace it, a reverse network will decide whether it is repaired, resold, stripped for parts, or dumped. That nested complexity is not a reason for despair, it is a call to literacy. When you understand how flows create civilization, you are better at improving them. Buying durable goods and favoring repair lengthens loops. Supporting standards and transparency helps small producers join big markets without being crushed. Voting for infrastructure that connects hinterlands to hubs equitably spreads opportunity. Backing fair labor and environmental safeguards ensures that gains are not made by hiding costs where you cannot see them. For leaders, the work is similar but scaled. Invest in ports, rail, and digital links that reduce inland isolation. Cultivate legal predictability so strangers can transact safely. Keep a few strategic inventories where substitution is hard. Train the translators, literal and figurative, who bridge cultures and industries, and diagram the network often enough that you know which links are critical and which can be rerouted before a crisis decides for you. Let the thought close gently. Every civilization you admire is less a fortress than a conversation carried by roads and winds, by friendships and ledgers, by ships and promises. When those conversations flow widely and fairly, cities brighten and strangers become partners. When the paths narrow or sour, wealth curdles into suspicion. You do not need to memorize the trade routes of the past to walk wisely in the present. It is enough to feel the grain of movement in what you touch and to choose in the small radius you control, to keep the channels open, the signals honest, and the gains shared. Because prosperity is not a hoard, but a current, and civilizations thrive when they learn how to let it move. Power grids, cascades, blackouts, and stability. Stand in a quiet room and listen for the faintest hum. You are hearing the heartbeat of a continent, a chorus of machines trying to stay in step. And once you learn to read that sound, you can feel what a power grid really is. Not just wires and towers, but a living balance between supply and demand that must be kept almost perfectly, even from one breath to the next. Because when more people switch things on than generators provide, the shared frequency sags. When generators push more than loads consume, it rises, and the entire system uses that tiny drift as its most honest signal of stress. In simple terms, thousands of generators, big steam turbines, gas units, hydropower plants, and more recently, solar and wind inverters, synchronize so their electrical waves crest together. The massive spinning machines provides inertia, a physical buffer that resists sudden changes, and old-fashioned governors on those machines nudge fuel valves when the frequency strays, adding or shaving power automatically in seconds. Above that, control rooms run automatic generation control, adjusting whole fleets every few minutes to steer the grid back to its target. Market dispatches and unit commitments shape the next hours. Long-term planning sets which lines and plants even exist. Stability is won or lost in the spaces between these layers. Transmission lines are designed with headroom so that if one fails, others can carry the load. A principle called planning for one contingency. A sober way of saying you assume at least one bad thing will happen, and you still must be fine. But reality is bolder. If a line trips from a storm or a brush with trees, its flow leaps sideways to neighbors. If those neighbors were already near their limit, they overheat and trip in turn. Protective relays, meant to save equipment, can become accidental amplifiers if their settings are too tight or their data stale. Within seconds, the stress can run across regions like a crack through ice, and lights fade. Not because one big thing broke, but because many small safeties acted in honest isolation. Cascades have anatomy you can learn. A disturbance starts, weather, a mechanical failure, a wildfire cut, then power redistributes, angles between areas widen, and currents climb. Voltage begins to droop because reactive power that props up voltage is local by nature and doesn't travel as easily as real power. So cities far from the original fault can slip toward a kind of fainting spell known as voltage collapse. Generators that sense trouble may trip to protect themselves, tightening the noose. If the frequency falls too low, under-frequency load shedding deliberately disconnects blocks of customers to save the core. If islands of grids separate and drift out of step with each other, they must be rejoined carefully or kept apart until they are steady. Recovery, when it comes, is another discipline. Black start units, plants that can start without external power, ignite first, they energize a path to the next units. Operators rebuild islands, match their frequency and phase, and then close ties like surgeons reconnecting vessels. The art is to add load and generation in lockstep, so the newborn grid does not stumble. The physics is not vindictive, simply tight. Electricity moves along all available paths, not just the ones you'd prefer, and flows grow in proportion to differences in electrical pressure between regions. Long lines face thermal limits and stability limits. Even a generous map can bottleneck at a single interface because steel and geography disagree with human boundaries. Modern change adds new chapters to the same book. Wind and solar bring clean energy and in many places lower costs, but their usual inverters are grid following. They lean on an existing rhythm rather than setting it, and they contribute little traditional inertia, so a large sunlit afternoon can be both powerful and delicate if a big plant trips and there is not enough spin to slow the fall. This is why you hear about synthetic inertia and grid-forming inverters. Control modes that make power electronics behave more like leaders than followers. Holding voltage and frequency steady the way a good drummer keeps time when the band wobbles. Demand can help too. Instead of only chasing load with generation, operators can nudge or contract with factories, data centers, and fleets of smart devices to trim or shift consumption in seconds. One smelter briefly lowering a pot line, 1,000 water heaters pausing for a few minutes, a city's pumps retiming overnight cycles. Each is a tiny governor in the fabric of the grid, and together, they turn customers into stabilizers rather than stress. High-voltage direct current links add another tool. They can ship large blocks of power long distances with precise control and, because they don't require precise phase alignment, they can bridge asynchronous regions or isolate disturbances, acting like fire breaks as well as highways. Yet technology alone cannot carry the discipline if institutions lag. Grids depend on shared situational awareness. Modern sensors called phaser. Measurement units watch the dance of voltage angles, dozens of times a second, and on data and models that are kept realistic as equipment ages and patterns change. They depend on vegetation management that looks boring until it saves a city. They depend on markets and rules that value fast response and local voltage support, not just megawatt hours over a month. Weather is no longer a background actor. Heat waves raise demand and weaken line capacity. Drought cuts hydro. Storms and fires force preemptive shutoffs. Icing, dust, or salt can make equipment misbehave. Planning for yesterday's climate leaves buffers too thin for today's spikes. Cybersecurity now lives beside hard hats. Malicious commands can mimic the early steps of a cascade if protections are not layered and communications not authenticated. Through all of this runs a practical idea you can hold. Stability is not the absence of change, but the ability to absorb it without losing the song. In the first tens of milliseconds, inertial and fast inverter responses buy breathing room. In the next seconds, primary controls restore frequency toward target. Over minutes, secondary controls and dispatch settle the system. Over hours and days, markets, maintenance, and fuel logistics reshape the stage. Because each band has a job, resilience comes from diversity, different kinds of generation, different geographies connected by stout wires, different services rewarded, so there is no single point of cleverness holding everything up. If you run a city or a plant, the lessons scale down cleanly. Know your critical loads. Arrange icelandable backup where it matters. Keep sensors honest and alarms humane, so operators see signal rather than noise on hard days. And practice black start for the circuits you own so you are not waiting for a distant hand. If you are a citizen, stability is not far from your reach. Efficiency reduces the standing stress. Flexible use during peaks lightens the hardest hours. And support for upgrades. Substations that can host inverters. Lines that relieve chronic bottlenecks. Trees that cool streets without threatening conductors. turns good intentions into physical margin. It is tempting to treat blackouts as failures of will, but most are failures of alignment. Protection set to the last decade's grid. Assets run closer to the edge than their owners admit. Weather a little stranger than the average. Communications a little messier than the test. Fix those, and cascades become rarer, not through luck, but through design. Let the thought close softly. That faint hum you noticed at the start is a promise made fresh every second by people and machines you do not see. And when it falters, the dark is not a verdict, only a pause while the system catches its breath. Tend the buffers, reward the fast helpers, keep the trees at a respectful distance, and the grid will do what living systems do best: adapt, recover, and, with a little care, keep the lights warm against the evening. Transportation hubs. Why delays ripple worldwide? You notice it first as a polite chime above you, a single gate agent's voice tilting a departure by 20 minutes. And yet, somewhere far beyond this concourse, a factory's loading bay will close an hour late tonight, and a ship will idle outside a harbor tomorrow morning. That is the quiet power of transportation hubs. Places built to concentrate flows so that movement is efficient on average but exquisitely sensitive at the seams. And once you look behind the announcements, you start to see how a small delay turns into a ripple that travels the world. A hub is a promise. Many spokes meet so you can swap planes, trains, trucks, or containers with the least total mileage and cost. And because assets are expensive, runways, cranes, gates, locomotives, crews, the schedule stitches them tightly, leaving just enough room to breathe if everything goes as planned. But breathing room is the first currency spent when weather thickens. When an inbound aircraft parks on a taxiway waiting for a gate, when a crane operator pauses for a safety check, when a crew times out by law after a long night. Each of those moments narrows capacity exactly where many routes converge, and the queues that form at hubs are not just lines, they are memory, storing earlier trouble and paying it out slowly into the day. You feel this in air travel most clearly. The metal tube that carries you at dawn is also this afternoon's flight to a different city and tonight's to a third. The same cabin crew may be scheduled to work the first 2 legs, a different team the third. A maintenance window waits at a specific base where parts and technicians are staged. The gate you occupy is reserved minutes later for an arrival with tight connections. The runway can clear a plane every small handful of heartbeats at best. And when visibility drops, spacing increases, slashing throughput. One 20-minute hold at a hub can break downstream connections for 100 passengers, divert cargo to a later belly hold, push a pilot over duty limits so a backup must be called from a hotel, and trap the next plane on the ramp with nowhere to park. You see a single delay. The schedule feels a missing stitch that must be resewn all day. Seaports have their own choreography. A container ship's berth is booked in a precise window that aligns pilots, tugboats, cranes, and yard space. A late tide or fog at the harbour mouth shifts that window, and the terminal's stacks, organized like a 3-dimensional crossword of colored boxes, must be reshuffled to reach the rightful container without moving half a dozen others first. A crane taken out of service adds minutes to every lift. A shortage of chassis or drivers strands boxes in the yard. Rail slots to inland depots back up if one train misses its path on a busy corridor. Because containers are also appointments at warehouses with finite doors and labor shifts, a day's slippage at the quay can turn into a week's drift in the hinterland. Replenishment orders pulled forward, truckers circling for new times, empty boxes in the wrong city when exporters need them most. Rail hubs carry their own constraints you can hear in the horn at midnight. A yard classifies cars into new trains by destination blocks. If one inbound arrives late, its cars miss the build and must wait for the next plan. On single-track segments, meets are scheduled like dances. One train must take the siding while another passes, and a single slow climb over a grade pushes the meet to the next siding, rippling across hours. Locomotives and crews are not interchangeable everywhere. Inspections and fueling are set at fixed points. Track maintenance windows close lines entirely for a spell, so the network absorbs trouble in pulses, not smoothly. On roads, the hub is often a cluster of distribution centers where appointments are booked to 15-minute windows. A late morning slot creates afternoon congestion that spills onto arterials, and driver hours of service rules mean a 30-minute wait can force an overnight reset. The common thread is synchronization. Hubs are efficient because many independent plans meet exactly, which means they are fragile exactly where those plans share a resource. Runway, gate, crane, berth, switch, dock door. Metrics can quietly worsen this fragility. If an airline is rewarded for on-time departures from the hub, it may push a flight to leave without a handful of connecting passengers, preserving a local score while exporting missed connections to the network. If a terminal is measured on moves per hour at the crane, it may pack stacks densely to reduce crane travel, only to pay later when a single out-of-sequence box forces costly rehandles. The physics is simple even when the systems look grand. When demand approaches capacity at a shared bottleneck, queues grow non-linearly. When variability rises, storms, labor shortages, health protocols, sudden demand, it exploits every tight spot. Recovery then is a craft. Schedules carry slack in hidden places, longer ground times on certain pairings, extra locomotives staged at busy nodes, reserve crews on call. But slack costs money, so it tends to be shaved in good years and missed in bad ones. Better designs make slack do double duty. Aircraft that can swap types without retraining entire crews, gates that can handle multiple models, data shared across competing airlines and the airport so that a delay at one gate can be absorbed by another, railroads running timetable precision so dispatchers can see conflicts hours ahead, trucking appointments that adjust dynamically when a storm slows a highway. Some solutions are physical, more taxiways to avoid gridlock, longer passing sidings, on-dock rail to decouple yard and road. Others are informational, live feeds from aircraft and ships, predictive arrivals that tell a crane team which box will be on top, collaborative decision-making rooms where port, line, and customs sit together and choose the least worst option in minutes rather than hours. And some are cultural, rewarding total trip on time instead of only segment punctuality, practicing disruption drills so that when a runway closes or a berth is blocked, the playbook is muscle memory, designing incentives so that a local win does not create a global loss. You finish this thought with a simple, useful picture. Hubs are amplifiers of speed when conditions are kind, of delay when they are not. And the ripples you feel are not fate but the honest output of choices about buffers, shared resources, and how we count success. If you are a traveler, a shipper, a planner, you can nudge that output by favoring connections with sensible layovers, by staggering deliveries, by building small reserves into your own promises. If you are a steward of a hub, you can publish bottleneck maps, align measures with journeys rather than single steps, and make flexibility a feature rather than an afterthought. Then the next time a gentle chime announces a delay, you will hear more than inconvenience. You will hear a living system, absorbing a bump, and you will know why. Sometimes the kindest thing you can do for the world's schedule is to let one flight wait 5 extra minutes for the last connection, because a ripple can carry harm or it can carry care, and civilization's routes are smoother when we choose the latter on purpose. Internet topologies, robust, scale-free, and surprising, follow the path your click just took, and you'll discover a map that doesn't look like streets at all, more like a galaxy of islands stitched by invisible ferries, because the internet isn't one network but a federation of thousands of autonomous systems that agree moment by moment on where to send your packets, and its shape is both robust and odd in ways that reward a careful look. At the large scale, a handful of very well-connected providers and exchange points act as hubs while countless smaller networks attach as spokes, a scale-free-ish pattern born from a simple habit called preferential attachment. Newcomers connect where many connections already are. So most nodes have only a few links, while a few have a great many. And that heavy-tailed structure helps random failures vanish into the background, yet leaves the system sensitive to trouble at the biggest hubs. Under the hood, the decisions aren't made by a central dispatcher, but by a conversation called BGP, a path vector protocol where networks announce the blocks of addresses they can reach and attach policies about who can use which paths. Policy matters because the internet is not a charity. Some links are paid transit, others are free peering between equals, and route choices reflect cost, contracts, and convenience as much as raw distance. So the shortest path by geography may still lose to a path that exits your provider's footprint sooner, hot potato routing, or that respects a partner agreement. Between these islands and policies live the places where the graph becomes a city square, internet exchange points, IXPs, warehouse-like facilities where hundreds of networks plug into a common fabric and swap traffic directly, shrinking hops and latency with a single cross-connect. Their growth has flattened parts of the internet, pulling flows off transcontinental backbones sooner and making local content feel local. Yet even as the fabric spreads, another layer has quietly centralized experience. Content delivery networks and large cloud providers have built massive private backbones and planted caches near the edge. They use Anycast, advertising the same address from many places at once. So your request lands at the nearest healthy site, and they steer traffic around faults with internal telemetry faster than the public control plane can react, which is why a video can stay smooth even when a fiber is cut somewhere you'll never hear about. Not all surprises are so kind. Because BGP trusts its neighbors by design, a misconfiguration or a malicious route hijack can make a network briefly claim addresses it doesn't own, dragging traffic the wrong way until operators notice and withdraw the lie. Defenses like RPKI add cryptographic proofs of origin, and route filtering at IXPs and providers has improved, but the system's openness means vigilance is part of its nature. The data plane and control plane move at different tempos. Packets stream along established paths in microseconds, while the routing conversation reconverges in seconds to minutes after a failure. During that period, you may see odd detours or brief timeouts as the map reshapes itself. Inside, big networks are different logic runs. Link state protocols such as OSPF or ISIS compute shortest paths, with fresh knowledge of topology. Segment routing and MPLS pin certain traffic to engineered tunnels, and equal cost multipathing splits flows across parallel links. So the internet's graph is really a patchwork of graphs, each stitched by slightly different rules, with BGP acting as glue at the seams. Physical reality still has the last word. Undersea cables bunch at hospitable landing sites, mountains corral fiber along valleys, and rights-of-way funnel conduits under the same bridges and streets. These constraints create choke points where one backhoe, storm, or earthquake can slice multiple carriers at once, reminding you that redundant paths aren't truly independent if they share a trench. The network's degree pattern also hides a rich club, a set of large networks that peer densely with each other. This creates path diversity and speed, but can concentrate influence, shaping which innovations propagate easily and which stall until the giants adopt them. Meanwhile, the naming system that lets you type words instead of numbers, the DNS, is both hierarchical and surprisingly resilient. Root servers are mirrored worldwide with Anycast. Resolvers cache aggressively, and modern stubs can try several upstreams at once. Even so, a misstep at a major registrar or a widely used DNS provider can ripple globally, proving that logical decentralization sits on a few operational pillars that must be tended with care. If this sounds precarious, the other side of the ledger is impressive. The internet's small world character, the fact that most nodes are only a handful of hops apart, means failovers often find another viable route. The mix of public and private backbones gives multiple ways around damage, and the habit of peering locally means your country can keep talking to itself even when a distant cable is down. Some of the most interesting dynamics are human. Capacity planners chase diurnal waves and sudden cultural spikes. Operators trade traffic blackholing communities during active attacks to dump malicious floods before they reach victims. And protocol designers have learned to hide new transports inside old shells. Witness Quick carrying modern features over UDP, because middle boxes ossified around yesterday's expectations. There are myths worth dropping as you learn this terrain. The internet routes around censorship is only partly true. Filters at key international links or pressure on a small set of domestic IXPs can contain traffic, and deep inspection can throttle unwanted protocols unless they disguise themselves well. Decentralized doesn't mean leaderless. The health of standards bodies, the openness of peering policies, and the operational culture at a few large actors shape the everyday topology more than a random drawing of nodes would suggest. Scale-free is not a law but a tendency. Measurements shift over time as IXPs spread, access networks grow, and enterprise traffic migrates to clouds. What stays consistent is the heavy tail, lots of small, a few huge, and the robust yet fragile trade-off it brings. The practical lessons for you are modest and real. Diversity beats wishful thinking. So 2 upstreams from different access paths protect a business better than more bandwidth on one. Secure the edges you control. Turn on DNSSEC validation at your resolver. Prefer providers that enforce RPKI. Monitor paths so you notice when your prefixes take a strange tour. And design applications as if the network will occasionally lie or vanish. Timeouts, retries, idempotent operations, and fallbacks turn a brittle user experience into one that rides through reconvergence without drama. Hold the closing image, a graph you cannot see, mostly stable because thousands of operators in different time zones keep their piece tidy, a handful of hubs that make the whole feel small, countless bridges that bring novelty across community lines, and a control conversation that never ends, updating, withdrawing, learning, proof that scale and surprise can live together when a system invites many honest voices to agree just enough on what reachable means and leaves room for the rest to improve quietly underneath. Supply chains, hidden links behind. Every product tug gently on any price tag, and you'll feel how much is hidden in that small strip of paper. The quiet truth is that most products are biographies written across factories, farms, ports, data feeds, and decisions you never see. And learning to read those biographies turns supply chain from jargon into a map of how things really arrive in your hands. You can start with the cast of characters, a brand that designs and promises, a first-tier supplier that assembles, and behind that, a staircase of second and third-tier specialists who make the subcomponents, the powders, the films, the screws. Each step adds lead time and risk, so planners try to place the right buffers in the right places. Raw materials stored near mines, critical chips held at assemblers, finished goods staged close to customers. Because where you keep the extra is as important as how much you keep. In a calm week, the system hums through a routine. Retailers send point-of-sale data upstream. Factories translate demand into work orders. Warehouses receive advanced shipping notices, so crews and dock doors are ready. Barcodes and RFID tag movements as cases are cross-docked or slotted for picking. Trucks and trains close the last miles under contracts that specify who owns risk at each handoff. Under the hood, the calendar has a heartbeat called Sales and Operations Planning, a recurring meeting where demand forecasters, production schedulers, finance, and logistics reconcile optimism with physics. Machines have changeover times, tools need maintenance, yields are never perfect, and labor arrives in shifts, not in abstractions. A simple but brutal dynamic haunts these conversations. The bullwhip, where small changes in store demand become large swings in factory orders as each tier pads for uncertainty, batches for efficiency, and reacts to promotions or shortages. The cure is partly cultural and partly technical. Share real retail data, stabilize order cadences, avoid end-of-month hockey sticks, and reward service level and total cost instead of just warehouse turns. Quality rides the same rails as quantity. Medical, food, and aerospace goods move under cold chains, clean rooms, and serialized traceability that can pinpoint which lot of resin became which batch of vials and which pallet left which room at which time. When something goes wrong, containment depends on that memory being honest, not heroic. Lot codes that can be read, audits that are more than paper, supplier approvals that go past the lobby. Compliance acronyms sound dull until you see their purpose. Hazard analyses prevent a contaminant from traveling. Process controls keep a small drift from becoming a costly recall. Failure mode reviews ask what if long before a part ships. Sourcing strategy is quieter but decisive. Single sourcing a clever part lowers cost and simplifies life until a flood, a fire, a strike, or a sanction arrives. Dual sourcing raises unit price but buys optionality. Geographic diversity lowers correlated risk. Nearshoring trades cheap labor for shorter lead times and easier visits. Postponement keeps a product generic, late into the flow, so color or region-specific features are added near the customer, cutting stranded inventory. The same product can be a triumph or a liability depending on where its decoupling point sits, the moment when it stops being a flexible, semi-finished good and becomes a specific item with a committed destiny. Logistics stitches it together with ordinary magic. A standard container lets a blender in one country ride a ship, a barge, a train, and a truck without repacking. Harmonized customs codes and certificates unlock borders. A chassis shortage at a port can ripple hundreds of kilometers inland by starving warehouses of empties. A winter storm that closes a single Highland pass can force milk to be dumped because it cannot wait for road salt. Control towers, really rooms of screens fed by EDI messages, API calls and satellite pings. Try to see these ripples early, but visibility is not the same as control. You still need pre-negotiated alternates, carriers you can call at odd hours, and contracts that allow rerouting without turning exceptions into lawsuits. Inside warehouses, design is a kind of choreography. Fast-moving items live near the front and at waist height. Robots or conveyors carry totes to human pickers. Wave planning groups orders. So one trip down an aisle fills many boxes. Slotting changes with seasons. Safety stock protects service, but too much creates dust and shrink. In factories, the physics of flow rules whether promises can be kept. Bottlenecks set the pace, not your wishes. Changeovers are paid for in hours and scrap. An andon pull at a lean plant halts the line to fix a small defect before it multiplies. Overall equipment effectiveness is less a trophy than a mirror about losses to downtime, speed, and quality. Digital experiments help, but hype is easy. A digital twin that mirrors inventory and lead times can test what if a supplier misses a week and show which orders to resequence. Predictive tools learn from weather, port congestion, and search trends to sense demand early. Yet dashboards without authority are decoration, and a blockchain without data discipline is just an expensive spreadsheet. Truth still begins with clean scans, disciplined master data, and people who trust the numbers enough to act. Risk and ethics run through the same veins. Scope 3 emissions, the footprint embedded in the goods you buy, live mostly in supply chains, not in headquarters. Conflict minerals, child labor, and unsafe mines do not disappear because a contract is signed 2 tiers away. Responsible sourcing means tracing beyond your first tier, paying for alternatives, and rewarding suppliers that fix root causes rather than polishing reports. Circular flows are the frontier that turns cost into value, designed for repair so failed items come home as refurbished stock, harvest parts from returns, recapture metals in batteries, closed-loop packaging so fewer pallets and films walk out to the landfill. Every closed loop shrinks the chain and thickens resilience. If you run a small shop, the same principles scale down. Pick 1 or 2 backup sources for critical items. Agree on minimum order quantities before you're desperate. Keep a shared living bill of materials with your assembler, and measure the outcomes that matter to your promises. Fill rate, lead time reliability, defect escapes, rather than flattering totals. If you're simply a citizen deciding what to buy, tiny habits bend the network. Choosing durable, repairable goods reduces churn. Buying seasonal or local where it truly lowers waste trims unnecessary miles. Rewarding companies that publish supplier lists and corrective actions helps honest actors compete with cut-rate opacity. The gentlest conclusion is also the most practical. Behind every product is a chain of links that can either vibrate with panic or hum with care. And the difference is made by 1,000 small choices. Where you place slack, how you share information, which risks you admit in daylight, and whether you design exits as thoughtfully as entrances. Learn to see those links and you stop treating shortages and gluts as weather. You start tuning a system whose job is not to be perfect, but to be dependable. And that steadiness is the quiet gift a good supply chain leaves in your home each time a box arrives exactly when you hoped. Financial networks, contagion in markets. Start by picturing a balance sheet not as columns of numbers but as a small map. On the left, assets that can be sold or pledged. On the right, promises that must be met, and then zoom out until thousands of these maps touch and overlap. Because a modern financial system is a network of mutual claims, and contagion in markets is what happens when stress on one map redraws the lines on many others faster than people can adjust. The simplest path is mechanical. A lender fails to pay. Its creditors take losses, their capital thins, their own creditors get nervous, and credit tightens in widening circles. But most cascades travel a subtler route, through prices, margins, and confidence, so that even without an outright default, a sharp drop in the value of a widely held asset forces leveraged investors to sell. Those sales push the price down further, and the fall ricochets across portfolios that never traded with each other directly. This is the fire sale loop. Assets marked to market reduce measured equity, risk models demand cuts, lenders raise haircuts or call for more collateral. Positions are trimmed into a falling market, depth vanishes, and a paper loss becomes a funding problem that becomes a solvency question. In short, liquidity and solvency talk to each other until they sound the same. Funding chains add their own fragility. Many institutions borrow short and lend long. Maturity transformation that works in calm weather, but invites runs when depositors or wholesale funders suspect trouble. In repo markets, a rumor or a price move can make lenders demand more collateral or refuse to roll financing at exactly the moment borrowers most need it. So a repo run is just a fast version of an old story. Collateral itself can be reused, rehypothecated. So the same bond supports several loans down a chain. When one link breaks, everyone downstream scrambles to replace what they thought they controlled. Payment and clearing systems sit at the core of these webs. They are built to settle billions of small obligations safely every day. But a delayed payment from one large node can trap liquidity in the pipes, forcing healthy firms to postpone their own transfers and spreading stress by timetable rather than by rumor. Derivatives concentrate and disperse risk at once. A central counterparty stands between buyers and sellers and collects margin to absorb shocks, which reduces bilateral exposures in the web. Yet if many members are long the same trade and the market lurches, variation margin calls arrive everywhere at once, and the scramble for cash can drain liquidity from unrelated markets. Clearinghouses are strong precisely because they are conservative about collateral, but in a system short of cash, the strictness that protects them can amplify demand for cash outside. The shape of the network matters just as much as its parts. A core periphery structure, few big hubs, many small spokes, can absorb random small failures easily, yet be sensitive to distress at a hub. Dense clustering among similar institutions spreads panic quickly within a club. Long funding chains that promise daily liquidity at one end and hold hard-to-sell assets at the other create hidden springs that release violently when pulled. Crowded trades add a human echo. When many funds rely on similar signals, use comparable value at risk limits, and borrow from the same prime brokers, their de-risking looks synchronized even if they never speak. So a price shock does not just change opinions, it forces the same action across desks. Procyclicality is the quiet villain beneath these patterns. In good times, rising prices lower measured risk, margins shrink, leverage grows, and funding shifts toward cheaper, shorter sources. In bad times, the direction reverses exactly when balance sheets are least able to accommodate it. Policies that seem prudent in isolation, tight stop losses, hard leverage caps, daily liquidity promises on portfolios of slow-moving assets, can collectively yank the wheel in the same direction. Against this tendency, safeguards try to add drag and buffers. Capital and liquidity requirements give banks skin and stamina. Liquidity coverage and stable funding ratios nudge balance sheets towards structures that survive a few hungry days. Stress tests make firms rehearse severe but plausible paths, so they discover where cash will be short before a real storm does. Living wills force large institutions to map their own critical functions. A grim exercise that pays off when a failure must be contained without freezing the rest of the market. Market-wide tools buy time. Circuit breakers pause trading when moves become disorderly, giving humans a moment to overrule reflex. Central bank discount windows and standing repo facilities let solvent institutions turn good collateral into cash without a public spectacle. Lender of last resort actions follow an old rule: lend freely against quality collateral at a rate that discourages casual use, because the point is not to reward errors, but to stop a solvent system from mistaking a liquidity shortage for a fatal wound. Transparency helps more than slogans. Regular disclosures on exposures, standardized reporting of securities financing, and public dashboards on market depth and margin conditions let participants see the same storm rather than imagine different ones. None of this eliminates risk, and it shouldn't. Risk is the price of allocating capital to new ideas, but it can redirect where risk lives and how it moves. Practical early warnings exist. Rising leverage funded by very short-term sources, growing overlap in the largest holdings across funds, shrinking market depth relative to the daily turnover of popular instruments, sharp, synchronized increases in margin requirements across venues, and the spread between secure and unsecured funding rates widening in calm news. When those dials twitch together, managers can switch from maximizing return to preserving function. Slow redemptions rather than promise daily exits from illiquid pools. Diversify collateral and counterparties before it's fashionable. Prefer term funding long enough to ride out a bad week. Run tabletop exercises that start with a major participant misses payment at 10:03. What do you do by 10:05? and give risk teams permission to be dull on sunny days. If you are an individual saver, the network lesson scales down cleanly. Diversify across asset classes and institutions. Match your own liabilities timing to your investment's liquidity. Be wary of products that promise safe and steady returns from opaque strategies funded with short money. And remember that the yield you can see often reflects a hidden promise someone else must keep under stress. For policymakers and leaders, humility is part of competence. Prepare for the next crisis to rhyme but not repeat. Watch the shadow banking periphery where new fragilities often incubate. And design rules that lean against the cycle rather than chase it. Counter-cyclical buffers that rise in booms and fall in busts. Margining that considers system-wide liquidity, and resolution regimes that make shareholders and certain creditors absorb losses before taxpayers are asked. The comforting truth, if you let it in, is that financial contagion is not a supernatural wave. It is the outcome of many small constraints interacting: funding calendars, collateral practices, shared models, and the topology of obligations. And that means many small, honest choices can blunt it. Keep buffers where they actually matter. Avoid building promises that can only be kept in fair weather. Reward firms for resilience as much as for quarterly speed. And as you set the thought down for the night, hold this last image. Thousands of balance sheet maps lit like a quiet city at dusk, each with its own flows and deadlines linked by contracts and trust. When one window darkens, the light may flicker next door, but with enough spacing, enough water in the cisterns, and neighbors who know the drill, the city stays lit. Not because fear was absent, but because design and discipline kept fear from finding a straight road. Neural organoids, mini-brains that learn. You meet the idea in a dish, not a cradle, a pale, pea-sized sphere of living cells grown from human stem cells swirls gently in nutrient media. And although it has no face, no blood, no body, it generates electrical whispers that change with experience. This is a neural organoid, a self-organizing tissue that models parts of the developing brain. And the honest wonder is that such a small assembly can display hallmarks of learning when you give it the right inputs and ask the right questions. Scientists begin by reprogramming adult cells back into an embryonic-like state and nudging them with timed cues so they take on neural identities. Left to themselves in 3 dimensions, the cells sort into zones that resemble early brain regions, sprouting neurons and support cells, forming synapses, and producing spontaneous activity that matures over weeks to months. These aren't tiny replicas of a mind. They lack the ordered layers, rich blood supply, and sensory context of a living brain. But they do recapitulate basic circuit rules well enough to become laboratories for development and disease. When placed on multi-electrode arrays, organoid spikes can be recorded and stimulated, turning a dish into a feedback loop. Regular patterns of input encourage specific networks to fire together. Irregular noise discourages unhelpful patterns, and over repeated trials the tissue shifts its responses toward the predictable stimulation. A simple definition of learning that emerges from synaptic plasticity rather than from any secret plan. Groups have shown that if you couple the firing of an organoid to a task with a simple goal, stabilizing a sensory signal, steering a controllable object on a screen, distinguishing between 2 input streams, the network's output improves with practice, and when you change the rules, it adapts again. Not because it understands, but because circuits that reduce surprise are favored as connections strengthen and weaken. Other organoids teach different lessons. Retinal versions grow light-sensitive cells that respond to flashes and transmit signals to downstream neurons. Midbrain-like versions make dopamine and reveal how particular mutations alter neuron survival. Assembloids fuse distinct organoids, such as cortical and spinal-like tissues, so that activity in one drives contractions of attached muscle fibers, a crude but thrilling way to study long-range coupling without ever touching an animal. Alongside these abilities sit blunt constraints that keep the story honest. Without blood vessels, oxygen and nutrients diffuse slowly, so organoids remain small and can develop stressed cores. Bioreactors and microfluidic perfusion ease some limits, and transplantation into rodent brains can recruit blood supply and extend maturity. But standardization is still a work in progress. Batch variability is real. 2 organoids grown with the same recipe may differ in cell mix and rhythm, so labs are building benchmarks, gene expression panels, firing statistics, responses to a common set of stimuli, to separate robust features from noise. Electrodes age. Glial cells reshape networks as they mature. Long-term stability is as much engineering as biology, and the word learning needs care. What is measured are shifts in firing and connectivity that align with a goal set by the experimenter, not comprehension. The closer you tie a circuit to clear feedback, the more reliably it adapts. Even within those limits, the uses are profound. Because organoids carry the donor's genome, they let researchers watch how a specific variant influences migration, synapse formation, or network excitability, often revealing a window for therapy that animal models missed. Drugs can be screened on human tissue at meaningful stages. Infections that target developing brains can be studied without risking a fetus. And closed-loop training regimes provide standardized stress tests for circuit resilience. A newer ambition, sometimes called organoid intelligence, treats these networks as living processes that can learn patterns with stunning energy efficiency compared to silicon. It is early days, but hybrid systems where organoids handle certain adaptive tasks and digital systems handle precision. Arithmetic are being explored, and the research pays dividends back into basic neuroscience by forcing tight definitions of input, reward, and performance. Ethics shadows the field because it should. Today's organoids lack the complexity thought necessary for feelings or awareness, and their behaviors can be explained by local plasticity and network dynamics, not by inner experience. Still, as tissues grow larger or are endowed with richer sensory coupling, labs are beginning to monitor proxies for integration and responsiveness, set limits on stimulus types, and design experiments to minimize distress even when distress is unlikely. Oversight committees that once focused on embryos and animals now include expertise in neural development and philosophy, and transparency about capabilities prevents hype from outrunning reality. The path forward is practical. Better vascularization and waste removal to sustain months-long maturation. Gentler, high-density interfaces that read and write without scarring. Standardized tasks that let one lab's result compare to another's. Open datasets of spiking and gene expression to refine models, and a culture that celebrates negative results so recipes improve faster. If you are listening for a larger meaning, take the quiet one. Organoids are proof that learning is not all or nothing but a property that begins as soon as excitable cells can change their connections in response to patterned input. In that sense, they are less mini-brains than windows onto early rules, how neurons become reliable partners, how networks settle into rhythms that carry information, how feedback sculpts function from possibility. Let that settle you rather than scare you. We are not growing minds in jars. We are building careful models that let us ask human questions in human tissue under human control. And with each carefully designed loop of stimulus and response, we learn how fragile development is, how generous plasticity can be, and how far thoughtful limits will take us. When you set the dish back on its cradle, and the incubator door closes with a soft seal, you leave behind no ghost, only a living tool that gets a little better at a small task every day, teaching us in return how learning itself takes root. Microbiome guilds, cooperations in your gut. You carry a rainforest inside you that runs on leftovers you don't digest, and its quiet prosperity depends less on star species than on work crews. Guilds that share tasks none can finish alone. Think of one group taking the first bite of complex plant fibers, another trimming those scraps into smaller sugars, a third fermenting the pieces into short-chain fatty acids, and yet another mopping up the byproducts that would otherwise stall the whole line, because in the colon success means passing the baton cleanly. At the front of this relay are primary degraders, such as many bacteroids and certain ruminococcus, that crack open stubborn polysaccharides from beans, grains, and vegetables. They expose sugars that neighbors can see, and in doing so, they feed the street as much as themselves. Second-line fermenters turn those sugars into acids like acetate and lactate. Then specialist butyrate producers, often from groups like Faecalibacterium and Roseburia, convert those into butyrate, a molecule with two simple virtues. It powers the cells lining your colon, and it nudges immune cells toward a calmer, regulatory posture. Propionate and acetate, the other major fermentation products, head to the liver and beyond, signaling through receptors on nerve and immune cells that tune appetite, inflammation, and metabolism. None of that requires mysticism, only the chemistry of fermentation matched to receptors your body already carries. Cooperation also runs through hydrogen, a tiny gas that builds up during fermentation and can choke the process if it accumulates. Methanogens and sulfate-reducing bacteria act as sink guilds, consuming that hydrogen and keeping the whole market moving, while other specialists turn lactate, useful in small amounts but harmful if it piles up, into butyrate, rescuing value from what could have become waste. Around this economy sits the city wall, a guild that maintains your mucus layer and shapes bile acids. Akkermansia and friends nibble mucin cautiously, stimulating your gut to renew it. Done well. The barrier stays thick and slippery, keeping microbes in the lumen and inflammation down. Bile acids arrive from the liver as detergents. Gut partners modify them into a broader palette that, in turn, signals back to your metabolism about glucose, fats, and the pace of bile production itself. Even vitamins reveal teamwork. Several residents synthesize vitamin K and certain B vitamins that you can absorb, the microbial equivalent of a corner bakery that shares its extras with the neighborhood. Because guilds are about jobs rather than badges, the cast can change while the play continues. This is functional redundancy. If one butyrate producer drops after a harsh week or a course of antibiotics, another may rise to fill the gap if the diet still supplies the right feedstock. That redundancy is why communities can look different yet perform similarly. And it's also why they become fragile when many roles go empty at once. Disruption teaches the point. Broad-spectrum antibiotics don't just thin headcount. They cut links between guilds, leaving sugars unprocessed, hydrogen unclaimed, and niches open for opportunists that fare better in the new vacancy. In that window, pathogens that tolerate oxygen or thrive on simple fuels can bloom, because the steady hand of colonization resistance, your resident community eating first, acidifying the space, consuming oxygen, and producing antimicrobials, has slipped. Diet swings act more gently but no less decisively. A fiber-poor, additive-heavy routine starves fiber-degrading guilds and pushes fermentation toward protein residues that yield ammonia, phenols, and other compounds you'd rather see in smaller amounts. Shift toward diverse fibers and resistant starches, and you feed the guilds that make butyrate and propionate, acidify the colon, and support a thicker barrier. Diversity of fiber matters. Different plants present different molecular puzzles, so spreading your sources across legumes, whole grains, nuts, seeds, and colorful vegetables fills more workbenches than relying on one favorite salad. Early life is a masterclass in guild assembly. Human milk carries complex sugars that babies cannot digest. They are gifts for specific microbes, especially bifidobacterium, that ferment them into acids that lower pH and discourage interlopers while producing metabolites that train the infant immune system to tell friend from foe. As solid foods arrive, guilds reconfigure to handle new fibers, and small exposures to soil, pets, and fermented foods broaden the playbook. Oxygen gradients create yet another layer. Near the gut wall, small amounts of oxygen leak from tissue. Early colonizers consume it, pulling the environment toward the anaerobic conditions preferred by fiber fermenters deeper in the lumen. When inflammation leaks more oxygen into the habitat, oxygen-tolerant microbes expand and the economy tilts, an ecological echo of how city streetlights change who comes out at night. Not all interactions are hugs. Cooperation and competition weave together. Many residents secrete bacteriocins, precise antimicrobials that target close cousins, curbing would-be freeloaders without spraying the whole block. Others compete for trace minerals like iron, starving pathogens that rely on iron harvesters. The net effect, when the network is healthy, is a many-voiced veto against invasion and a steady production of molecules that your own cells read as things are under control. Science aims to recruit these principles without overselling them. Fecal microbiota transplants can reseed a collapsed community in a few specific conditions, but they are not a cure-all. Probiotics are most convincing when they replace a missing guild or provide a temporary service, like breaking down a particular sugar, rather than when sold as panaceas. Prebiotics, fibers designed to feed the right crews, make the most sense when they complement what your plate already supplies. Fermented foods add a different benefit. Live cultures pass through and leave signals that nudge residents or your immune system. Even if those visitors don't stay, they can instruct the locals for a while and broaden the chemical conversation. Circadian rhythms touch this world too. Your microbes cycle with your eating window. bile flows in daily waves, and late-night grazing can push activity into times when repair should dominate. Regular mealtimes help the guilds anticipate supply the way a bakery schedules proofing before dawn. If you want a practical way to care for this economy, feed it like you would feed a team you respect. Steady, varied fiber inputs, occasional fermented foods. Enough protein for you, but not so much that it becomes the main substrate for fermentation. Spices and polyphenol-rich plants that select gently for helpful guilds, and space between antibiotic courses, unless they're truly needed, with a plan to restore what was lost if you must take them. Curiosity helps too. Notice how your system responds to beans after a few weeks of gradual introduction versus a single bold serving. Watch how stress, sleep, and movement affect your gut. Nerves and microbes converse through hormones and immune mediators, so calming one often steadies the other. Let the philosophical lesson land lightly. Cooperation is not a slogan in your gut. It's physics and chemistry organized into neighborhoods that turn waste into fuel and uncertainty into steady work. When you choose what to eat, you are voting for which guilds get paid, which tools are kept sharp, and which signals reach your own cells about danger or peace. The reward for learning this isn't a complicated regimen, but a friendlier routine. You stock the pantry for helpers who pay you back with energy, a tighter barrier, and immune signals that settle rather than flare. And in return, your small ecosystem does what the best communities always do: make resilience feel ordinary. Bioelectric patterning, voltage maps, that shape limbs. Lay a fingertip against your forearm and imagine that beneath skin and tendon there is not only tissue, but a faint electrical weather map. Patches a little more negative, borders a little less so, because one of the most startling truths in developmental biology is that growing and healing bodies use bioelectricity as a patterning language, with cells setting and reading membrane voltages to decide where a limb begins, how many digits appear, and when growth should stop. This isn't the dramatic lightning of neurons firing, but a quieter, steadier current produced by ion pumps and channels, shared through gap junctions so neighborhoods of cells can hold a collective state, a kind of tissue-level memory that outlives any single cell. At the scale of a limb bud, embryos generate measurable voltage gradients across the mound of cells that will become an arm or a wing. Early amphibian and chick studies used microelectrodes and voltage-sensitive dyes to map those fields, and when researchers subtly shifted the electrical state by blocking gap junctions or altering the activity of ion channels, the number or placement of digits changed, not because electricity overrode genes, but because voltage is one of the signals that genes listen to, funneling into second messengers like calcium and cyclic AMP that open or close developmental programs. The actors are ordinary but arranged with intent. Sodium-potassium pumps and proton pumps establish a baseline. Potassium and chloride channels set how excitable or quiet a patch will be. Gap junctions made of connexins let groups of cells share ions and small signals, knitting them into a consensus. And voltage differences affect which transcription factors enter nuclei, how chromatin is packaged, and which growth factors get secreted or ignored. The proof moves from maps to interventions. In frog embryos, artificially hyperpolarizing small clusters of cells, making their inside more negative, has been enough to kickstart eye or brain-like structures in new locations, as if the voltage state marked, "You have permission to build this here." In regenerating frogtails, suppressing a proton pump stalls regrowth, while restoring the charge pattern restarts it. In planarians, brief blockade of gap junctions produces two-headed animals that keep regenerating that way, long after the drug is gone. A striking hint that body plan can be stored as a long-lived bioelectric circuit, rather than a fleeting chemical swirl. Limb biology adds a humbler but crucial detail. Wounds create injury currents, endogenous electric fields that exit the cut surface and re-enter surrounding skin. And many cell types, including progenitors important for repair, exhibit galvanotaxis, migrating directionally along those fields. Corneal and skin models show that steering or boosting these fields can speed and organize healing. And in bone, clinical stimulators that deliver weak currents or fields have long been used to promote union, suggesting that tissues are not merely sensitive to voltage, but built to use it during repair. The pipeline to application is already real. A silk-based bioreactor worn briefly on freshly amputated frog limbs, delivering a small cocktail of ion channel and growth-modulating drugs, led over months to leg-like structures with bone, nerves, and toes more elaborate than untreated stumps. The device's initial job, researchers argue, was to set the bioelectric and biochemical weather just long enough for innate programs to take over, proof that early cues matter more than long, heavy-handed interventions. Clinical genetics backs the principle from another angle. People with mutations in certain ion channels or gap junction proteins, like KCNJ2, a potassium channel, or GJA1, connexin 43, can have limb and craniofacial anomalies along with electrical symptoms, implying that when bioelectric patterning tools are disrupted, shape as well as rhythm suffers. The limb is not a passive passenger of genome and mechanics. It is an electrical project as well. None of this means voltage alone draws a hand. It means voltage is a coordinate system that talks to morphogens and mechanics, gating when and where they matter. Cells at the anterior edge of a limb bud may be slightly more polarized than those posteriorly. That difference biases which genes read signals like SH or FGF as meaningful. As cells divide and rearrange, gap junctions maintain local coherence, so that a finger territory keeps acting like one, even as its members change. The feedback runs both ways. Strain on tissue can affect ion channel expression. Metabolites alter membrane potential. Hormones tweak pumps. So bioelectric maps are not static posters, but living weather that receives and sends news. The tools for seeing and writing this language are catching up fast. Voltage-sensitive fluorescent proteins report subtle changes in living embryos. Genetically encoded calcium indicators reveal when voltage has opened gates for the next messenger. Optogenetic actuators let experimenters flip the sign of a patch of tissue for minutes and then watch fate choices change. Microelectrode arrays and scanning probe methods map injury currents as they heal. And computational models, once brittle, now integrate ion flux with gene networks to predict which manipulations will produce extra digits or cleaner joints. There are cautions worth keeping close. Bioelectric patterning is context-dependent. The same channel tweak that supports growth in a regenerating limb might encourage proliferation in a precancerous field, and indeed depolarized states in some models correlate with tumor-like behaviors. While restoring polarization can push cells back toward normal programs, the point is not to worship voltage, but to respect it as a powerful dial. Batch variability in organ and limb models is real. Measuring and standardizing electrical states across time is hard, and because channels act in families, drugging one often nudges several, so off-targets must be tracked with the same care you'd give a hormone or a morphogen. Yet the practical advice that emerges is surprisingly human-scaled. If you're designing a therapy for better healing, consider whether you can set the early map. Apply a small, well-shaped field at the wound. Deliver channel modulators briefly to restore a lost gradient, or use a scaffold whose piezoelectric fibers generate tiny currents as a patient moves. If you're building a limb on a chip to study birth defects, include conductance and coupling as first-class variables, not afterthoughts. And if you're thinking philosophically about how bodies remember shape, give yourself permission to swap the metaphor of a static blueprint for that of a circuit that stabilizes a pattern until it needs to change. What settles as you breathe is quietly liberating. Limbs are not merely welded by genes and pushed by mechanics. They are negotiated by cells whispering in voltage about where edges should land and how growth should taper. And that whisper can be heard, mapped, and, with care, rewritten so that future surgery, rehabilitation, and regenerative medicine might look less like force and more like tuning, guiding a patient's own tissue to recall the form it once knew how to make. Chemical signaling fields, diffusion as language. Breathe in and consider that the world around you keeps up a constant, quiet conversation in molecules, whispers that slip through water and air, drift through soft tissues, and fade with distance, and that you, down to your smallest cells, are fluent in this language of diffusion even if you've never named it. Because to live is to release a little signal, to sense a little gradient, and to decide from a faint slope which way to turn. Diffusion sounds abstract until you imagine a dye dropped in a still pool. The color spreads because jostling molecules wander randomly, and crucially, they do not spread linearly with distance but sluggishly. Farther takes disproportionately longer. So chemical messages are best at local news unless something concentrates or protects them. You can read the rules of this language through the problems it solves. A single yeast cell seeking a mate doesn't know where away or toward is, so it samples the density of pheromone across its own surface. If one side of the membrane sees a touch more than the other, receptors tip the local machinery to grow a bud in that direction. And if the difference is tiny, the cell averages longer to be sure. Because diffusion is noisy and wise, listeners linger. A bacterium in open water adds a different trick. It is too small to detect left-right differences, so it compares now with a few seconds ago while it swims. If the concentration improves, it keeps going. If not, it tumbles and tries another heading. Temporal comparison turns a blurry field into a compass without ever drawing a map. In soils and tissues, signals rarely travel alone. They bind to surfaces, get nibbled by enzymes, and are absorbed by cells, which means a field is not just a fog, but a dynamic balance of secretion, drift, and sink. You can make that concrete by picturing a wounded fin or fingertip. Damaged cells release a pulse of hydrogen peroxide that diffuses outward and is consumed by neighbors. Immune sentinels detect the shallow slope of that molecule and crawl inward along it. The slope's steep enough to guide, but not so steep that cells pile up at the edge. Remove the sink, or flood the tissue with background peroxide, and the trail goes flat, proving that a field exists only when sources and sinks shape it together. Some fields don't just drift, they pulse. Slime mold cells separated on a dish relay bursts of a small messenger, often camp. Each burst triggering neighbors to release their own, because each cell also breaks the messenger down. What propagates is not a rising tide, but a wave, high, then low, then high again, sweeping across the colony and choreographing a collective march without a conductor. In your nose, diffusion meets turbulence in a trickier conversation. The scent from a distant flower or kitchen doesn't arrive as a smooth gradient, but in patchy filaments shredded by air currents. Insects solve this by surging forward when their antennae catch a patch and casting crosswind when the signal vanishes. A simple policy tuned to a field that is intermittent by nature. Cells and tissues extend the vocabulary further by reshaping the stage. Cilia on airway surfaces stir a thin layer of fluid so that antimicrobial peptides reach every corner. In the gut, a gel of mucus slows diffusion of irritants while allowing nutrients to pass. In the brain's extracellular space, a crowded meshwork makes movement anisotropic. So what seems spherical in a textbook resembles a squeezed plume in reality. Because diffusion is slow at distance, organisms deploy couriers when they need speed. Blood carries hormones quickly over long ranges, but upon arrival, those molecules still spread the last micrometers by diffusion to their targets, handing the message from a fast highway to a slow whisper with no loss of meaning. The physics of this language imposes limits that life has learned to respect and exploit. Imagine trying to estimate concentration from rare arrivals at a receptor. Molecules come in clumps by chance, so a cell reduces uncertainty by sampling longer or by spreading sensors over a larger patch. This is why some neurons and immune cells build microvillian folds. More surface means more chances to catch faint cues, and why many receptors cluster into nanodomains rather than floating alone. Your cells are statisticians, averaging noisy arrivals until the confidence is worth acting on. The rules also imply trade-offs for engineers and doctors. A drug delivered as a depot under the skin creates a local high that decays outward. If the payload binds strongly to tissue, the front crawls, and the radius of influence stays tight. If it degrades fast, the peak is sharp, but brief. If it's lipophilic, it hoards in membranes, changing how far and how evenly it spreads. Controlled release devices are, at heart, diffusion grammars written in polymers and pores. Tumors rewrite the grammar to their advantage by generating steep gradients of oxygen, acidity, and growth factors. Those gradients summon blood vessels and shield resistant pockets deep inside where drugs reach poorly. Reverse the slope by normalizing leaky vessels or cycling pressure in the interstitium, and the same dose can penetrate more evenly. Microbes answer in kind, in a laboratory plate that gradually increases antibiotic concentration from one side to the other. Migration and mutation create a visible staircase of adaptation that marches along the diffusion field, a cautionary picture of how tolerable gradients, not just high peaks, can foster resistance. The tools to see and sculpt fields have grown gentle and precise. Chemists uncage signaling molecules with a flash of light, writing temporary dots and lines of attractant in living tissue to test how cells pass geometry. Microfluidic chips deliver stable, shallow slopes that let researchers measure the smallest difference a cell can detect and how long it must measure to do so. New dyes and genetically encoded sensors report not only the presence of a messenger, but the tiny local shifts in pH, calcium, or cyclic nucleotides that encode a cell's answer. When diffusion meets reaction, patterns bloom without a plan, spots and stripes in skin, spiral waves in heart tissue, patchy flourishes in microbial colonies. But you don't need to invoke grand theories to use the principle. Pairing a short-range activator with a longer-range inhibitor creates an annulus of just right, and flipping the ranges flips the pattern. In the clinic, pairing a growth cue with its dispersing brake can focus repair where it's useful and prevent overgrowth at the edges. Ethics and realism keep the ore from turning into magic. Chemical fields are one language among several. Electricity, mechanics, and direct contact all speak at once, and the meaning of a given molecule depends on context, concentration, duration, partner cues, and the history of the receiver. What reads as "come here" to a young cell may read as "differentiate" to a mature one. What is nourishing in one niche is inflammatory in another. Change the sinks and the same secretion becomes noise. Yet once you see diffusion as language, practical habits fall out. In tissue engineering, you don't just seed cells and hope. You design source points, choose scaffolds that bias spread, place enzyme sinks so fields shape a structure rather than blur it. In regenerative care, you time doses to the half-life of the cue and the pace of clearance rather than to the clinic's calendar. In everyday life, you accept that nutrition and microbiota signals profile the gut's chemical weather and that consistency. The slow, repeated input that diffusion favors often beats spikes for the outcomes you want. Let the idea close softly, from plankton sensing nutrients to a healing fingertip calling helpers, from a moth surfing a ragged plume to a lab-grown tissue taught to assemble by a staged release. The world works because molecules wander, and because living things have learned to treat that wandering as speech. When you picture fields not as fog, but as careful gradients carved by sources and sinks, uncertainty turns into usable guidance. And you can trust that most of what needs saying in the body and beyond can be said cleanly at close range and repeated patiently until even the faintest listener knows which way to go. Swarm robotics, machines that borrow nature's rules. You notice the charm first in how little each robot knows. A palm-sized rover that sees only a meter ahead, a quadcopter that can keep altitude and avoid the nearest wall, a puck with 2 wheels and a ring of cheap infrared sensors. None of them is clever alone, yet when you let hundreds loose with a handful of simple rules, patterns appear that feel less like choreography and more like weather you can steer. That is the working heart of swarm robotics. Machines that borrow nature's rules, local sensing, short messages, and feedback through the environment to solve problems too large or too messy for a single brilliant machine. The science begins with constraints you can trust. Communication in the real world is lossy. Bandwidth is scarce, GPS drops in forests and factories, and centralized planners choke on delays and scale, so a swarm pushes decisions to the edge. Each unit runs the same small program: follow neighbors without colliding, keep a preferred distance, react to gradients of a signal, switch tasks when local cues cross a threshold. And because every robot applies these rules at the same time, group behavior emerges without a leader. You've seen animal versions of these rules. Birds align and keep spacing, fish school, ants lay and follow scent. The robotic analogs are deliberately plain. Alignment comes from reading a few neighbors' headings. Spacing from simple repulsion when proximity spikes. Cohesion from a soft pull toward the center of nearby peers. And the scent is a virtual pheromone that can be painted as light on the floor, as projected patterns from above, as radio beacons, or as numbers written to a shared digital map. Robots climb or descend those fields the way bacteria and insects do. But with physics, you can tune. Because rules are local, swarms scale well. Adding 10 more units usually makes coverage faster and failure less likely. And when one robot dies, the rest root around the absence as if it were another obstacle. Division of labor, a subtle strength in social insects, has a practical code version. Response thresholds. Give every robot a sensitivity to certain stimuli, uncollected samples, unexplored frontiers on a map, littered debris, and the ones with low thresholds self-select into that task first. As local cues fade, those robots drift back to idle or to another job, and a balance forms without a scheduler. The same principle supports foraging, distributing robots to where reward is higher. Collective transport. If enough neighbors push the same heavy object, keep pushing. If not, recruit. And adaptive patrolling. Spread out when alarms are quiet. Cluster when an area looks busy. Coordination doesn't require chatter so much as consistency. Simple consensus schemes let robots average their neighbors' estimates of, say, a gas concentration or a boundary's position. Gossip protocols mix those averages until the whole group agrees to within a small error. And when strict agreement is too slow, quorum rules. If 5 of your nearest neighbors vote yes, switch. Settle choices like go left around the obstacle, quickly enough to keep motion smooth. Mapping and search look like magic until you name the parts. Frontier exploration asks each robot to head for the closest frontier, the edge between known and unknown, while avoiding collisions. Local occupancy grids built from LiDAR or camera are fused only with nearby peers to save bandwidth. Occasional rendezvous with a few different teammates blends maps across the group, and a trick called loop closure, seeing a familiar scene from a new place, tightens drift without a headquarters. When GPS is absent, relative ranging with ultra-wideband radios, visual odometry from a small camera, and periodic look-at-me markers keep teams aligned. When line of sight breaks in smoke or rubble, robots can drop tiny relays that knit a temporary mesh so messages hop toward the edge. Construction with swarms uses still another rule, stigmergy, the idea that work leaves traces that guide the next step. A brick placed at a certified location becomes both product and signal. Templates etched or projected onto a surface create legal sites where modules can latch. Cubic robots with magnets can climb and reconfigure based on nothing more than local pattern matches. And because no single unit holds the blueprint, a partial collapse doesn't erase the plan. The next arrivals read what remains and continue. Agriculture borrows the same toolkit. Many small rovers weed between rows using simple vision. Drones scan canopies for stress. A few seeders recalibrate paths when soil compaction or rain erases marks. Each machine reports a sliver of state, and the field stays productive without a giant, fragile harvester. Environmental swarms skim coastal waters or lakes, each boat following a local gradient of salinity or pollutant and keeping distance from neighbors. Together, they trace plumes far faster than one vessel zigzagging alone. None of this works unless you respect the unromantic parts: energy, safety, and interference. Swarm batteries deplete asynchronously. A practical rule is energy homeostasis. If your charge falls below a threshold and nearby coverage stays above a minimum, peel off to dock. If too many leave at once, the remainder raise their thresholds automatically. Docks themselves can signal availability, so queues don't form. Safety is enforced with invariants baked into each controller. Never exceed a closing speed relative to a neighbor. Maintain a no-go bubble around people and sensitive assets. Drop thrust or torque if sensors disagree beyond a bound. And fail dark, stop and broadcast distress, rather than improvise when localization fails. Radio contention can sabotage the best algorithm. Swarms avoid chatty all-to-all links by using short, periodic beacons, compressing messages into tiny summaries, and relying on the environment for coordination whenever possible. Light, tags, chalked grids. So radios carry only exceptions. There are honest limits and failure modes worth naming. Local rules can create deadlocks at doors and narrow passages. Designers break symmetry with tie breakers. Yield if you have higher ID or if you arrived later. Or they inject small randomness so jams melt. Over-eager positive feedback. Too many robots piling onto a hot task wastes capacity. Soft caps and local penalties. If more than 3 helpers are within arm's length, choose a different frontier. Flatten spikes. Real ground is not a lab. Dust blinds optical flow. Specular flaws confuse LiDAR. GPS multipath lies. Robust swarms lean on redundancy. 2 weak sensors that disagree honestly beat one perfect sensor that fails rarely but catastrophically. And they practice outside early so the pretty demo code grows calluses. Verification and ethics are not afterthoughts. Formal tools can prove non-collision and connectivity guarantees under stated sensor ranges. Scenario generators stress swarms with wind, blocked aisles, and a missing dock. And logging at the edge, tiny black boxes on each robot. makes post-incident analysis more than guesswork. Privacy and social license matter when 100 flying cameras arrive over a town. Good practice sets altitude and geofences, disables high-resolution recording unless explicitly needed, and uses on-device detection that shares counts or alerts, not raw video. You may wonder why we bother with many small robots when we could build one mighty machine. The answer is practical. Failures are independent, coverage is parallel, entry cost per unit is low, and tasks like under-canopy mapping, warehouse inventory, or indoor search have too many corners for a single body to reach efficiently. The philosophical turn is gentle. Swarms replace brittle perfection with graceful sufficiency, trading maximal individual capability for collective reliability. They make good enough everywhere soon beat perfect somewhere late. If you want to think like a swarm designer, you stop writing master plans and start choosing local contracts. What each unit must promise neighbors, what the world will look like when a task is done, and which simple sensations carry the news. And if you are simply curious about where this goes next, watch for 3 convergences that will make the field feel less like research and more like infrastructure. Edge learning that lets rules tune themselves while preserving safety bounds. Heterogeneous teams where air, ground, and water robots exchange roles. Drones scouting and dropping relays. Ground units doing heavy manipulation. Boats sampling and backhauling power. And human swarm interfaces that act like tending a garden. Set densities and no-go zones. Nudge fields. Read health at a glance, instead of piloting 100 toys. Let the thought settle softly. You don't have to control everything to get what you need. You can lay down a few honest rules, give simple agents the senses to follow them, and trust that order can rise from locality. And in a world of fragile complexity, there is comfort in machines that succeed the way good neighborhoods do, by watching their small circles closely, helping when the sign is right, and leaving clear signals for the next helper who arrives.